Ma'am did her graduation from University of Delhi and then her post-graduation, MPhil and PhD from Jawaharlal Nehru University, New Delhi. Her research interest includes abiotic stress, that is cold stress, functional genomics, proteomics, nitric oxide signaling, and nanobiotechnology. Ma'am has graded more than 10 PhD students and 15 MPhil students with more than 90 publications to her credit. Professor Renu Deswal is member of editorial board of several international journals like Frontiers in Plant Physiology and Journal of Proteomics, Proteins and Proteomics. Ma'am has edited several books and Ma'am has edited and co-authored several books, and she has she has been member and reviewer of various national and international grants like DBT, DST, CSIR, DRDO, National Science Foundation USA, Discovery Grants Canada, and many more. Professor Renu Deswal has organized several branch proteomics workshop and training program at Department of Botany, University of Delhi for research scholar as well as for faculty members. Ma'am has been associated with several professional bodies like Academic Committee member, Central Institute of Medicinal and Aromatic Plant, that is CMAP, Lucknow, India. She is chairperson, Development Committee and country representative of International Plant Proteomics Organization. Welcome, ma'am. Welcome to our conference. It is great to have you here. And over to you, ma'am.
Thank you, Ruchi, for the kind introduction. Can you hear me? Am I audible?
Yes, ma'am. You are
Okay. Somehow the camera is not working, but anyway, can you see my slides? I've shared them.
Yes, ma'am.
Okay.
We can see your slide as
So
well.
I think we can start with the lecture. I
Yes,
would
ma'am.
try to stick myself within 45 minutes, but probably I might exceed few minutes. So very very many congratulations to all of you for organizing this conference and workshop on omics. And this is the second day of the workshop, so congratulations to all the organizing member,
Thank you, ma'am.
and a very big thanks for giving me this opportunity to share a part of our work, what we are doing at Department of Botany. And as you see the title of the presentation, Protein Corona, an important tool for understanding nanoparticle biointeractions for safe and informed phytonanotechnology. So there are few words or few terms in this title, like protein corona, biointeractions of nanoparticle with the living system, and phytonanotechnology. So I would try to detail these terms to you. Actually, this day, as I gone through your program, what I could see was that this is a proteomics day for the workshop and the conference. So this what I'm going to present is also proteomics, but the next level. It's basically nanoproteomics, what we are doing now, and we are trying to use to answer some biological questions, trying to understand biology better using proteomics tool. So I'll just so what I've done, I've kept few slides on proteomics also, because it was a proteomics day for you, or you are basically learning more of proteomics on this day. And in the afternoon session, Dr. Ravi would be there. So I'm sure he will talk more technical things. It's not that I'm not going to talk technical, but what I'm trying to do is I'm trying to introduce, especially the students, the young generations, to this very fascinating and new area of research on nanoproteomics and how, excuse me, actually I'm not keeping well. So probably in between I have some problems of coughing and all. Bear with me for that. So when you talk of proteomics, genomics to proteomics, normally, or why there was a need? This is a big question. What is the need of newer areas like proteomics? So this slides gives, to some extent, it explains why there's a need of proteomics. Actually, the regulation which happens, this slides basically shows you the central dogma: transcription leading to translation. And at in between these points, there are regulatory points, like you have even transcription is controlled, then you have post-transcriptional controls, which would basically decide on RNA stability and even RNA processing. And finally, once this whole process takes place in the two-site and continuum, nucleus to cytoplasm, and then translation basically happens in your cytoplasm. And after translation, once the protein is translated, so not all messages are translated, because those post-transcriptional regulation, they'll control which messages would go, the stable ones would go for translation. And once the protein is formed, then you have PTMs, which would modify protein in different forms. So actually, what I'm trying to say is that the number of transcript would be few, or the genes which are leading to production of protein would be few, but number of proteins would be in huge numbers. Just to give you an example, when you talk of Arabidopsis, which is a model plant system, the genes are about 25,000, but the proteins are in huge numbers. Even in Arabidopsis, they are in thousands of numbers. So this variation, this happens because of all these modifications which are happening post-transcriptionally and post-translationally. And the credit of this term was coined by Professor Mark Wilkinson, who is an Australian scientist in the University of New South Wales. He defined this term, or he coined this term, proteome, and that's how this word, it started being used for analysis of protein in bulk. So this is how the outline of my presentation would be. First few slides on proteomics, then a bit on nanobiotechnology or nanophytotechnology, then coming to nanoproteomics, and in the end, protein corona. Little amount of work which we are doing on protein corona at botany department. We have initiated this work just 2-3 years back. Excuse me. And as I mentioned, this is a very new area of work. Not many people, not especially plant people, they are not working, many of them are not working on protein corona. So I just want to introduce this exciting area to all of you, what it is. So when you talk of proteome and proteomics, proteomics is a study of proteome. This all of you would be aware of. And the full complement of organisms, it could be unicellular, multicellular organism, that is your proteome. Okay, and the study of it is proteomics. Normally there are two definitions. One is a classical one and second one is an inclusive one. Classical basically deals with the identification of proteins in bulk. Okay, but inclusive deals with inputs from different areas. Like you will have, besides identifying protein, you would try to understand their function, and there you take input from genomics, you do interactions, and you take input from there, you take even input from your messenger RNA information, and that is how you try to complement the information so that you reach closer to the functions of protein. And when we are analyzing our proteomes, we can get information about, we can characterize protein, we can understand protein interactions, and we can also identify various markers by doing proteome analysis. So this is about a bit about proteomics, and this is, I think, the last slide. This is just to give you a overview. So whenever you are doing this proteome analysis, this is the usual workflow which you follow. First step is your extraction, and tissue could be either, it could be a tissue or a unicellular cell, or it could be an organelle. The source of protein can be anything, depending on what your question is and what you want to analyze. So Once you've done extraction, then this extract is separated, or the proteins are separated, and normally there are two ways, either gel-based separation or liquid chromatography-based separation. And better separation, better resolution of results you get. So better results you get. So it's a very important step, separating these proteins on gels or by liquid chromatography before going for digestion. The next step is you trypsinize your proteins, and there's a need of trypsinization because our MS machine usually they identify peptide better, and they can help in identification of protein. So once we have done trypsinization, then these samples are fed into the MS machine. The MS machine then give you identification and also quantifications of protein. Now, switching gear, nanotechnology. Again, this is a buzzword, and I'm sure you must have heard about it, and many of you would be aware of it. So basically, when you talk of nanotechnology, it's the word nano. So the important point is your whatever matter you are using, whether it is as atomic scale or molecular scale, it should be in nano range, at least in one dimension, 1 to 100 nanometer. And nanoscience basically is the study of this nanomaterial. And nanotechnology is basically designing, characterizing, then production and application of structures which are at nanoscale. And you use their properties. Because once, and this is just a picture to give you a relative or relationship between sizes at nanoscale. A water molecule is of zero 0.1 nanometer, and a virus is about 100 nanometer, bacteria about 1000 nanometer. So this, and these, even these things we cannot see by eye, you have to use microscopes. Okay, and even human hair, the diameter of human hair is about 100 nanometer again. So just to give you a feel of the dimensions, this slide is just to tell you that. And nature, in nature also we have formation of nanoparticles, like wherever there's forest fire, even when you're cooking our food, heat is generated. Excuse me. So even there, nanoparticle formation takes place. Similarly, at site of volcano eruptions, then at the site of mineral deposits, because of pressure, high pressure and temperature, you have formation of nanoparticles. Besides, so this is all nature or naturally existing NPs or nanoparticles, but you can also synthesize them using extracts from various organisms, okay, including plants, and that is called green synthesis. So there, this is naturally existing, and this is basically your engineered nanoparticle, okay. And this engineering can also be done two ways, either chemical based or green synthesis. Green synthesis, whenever you're using extract from any life form, then it becomes green synthesis, like you are not adding any chemicals from outside to make the nanoparticles. When you're using chemical, that is your chemical synthesis. Now, what is phytonanotechnology? Phytonanotechnology, again, the term indicates itself. Whenever you're using this nanomaterials or nano-designed materials for on plants, it is called as phytonanotechnology. And there are various applications now. In fact, the literature is flooded with examples of various nanoparticles being used for sustainable agriculture. Why sustainable? Because especially if you are using green synthesized nanoparticle, you are not introducing any chemical into the environment. Okay, green synthesized nanoparticles are considered benign comparatively. Okay, so we have examples, many examples in literature about these NP working as nutrient carrier for gene delivery, as biosensors, as nanopesticides, as nanofertilizers. And so these are various applications which you'll find. So what has happened due to this? Now, tons and tons of these nanoparticles, they are present in our air, soil, as well as water, besides being present in biological systems, because we are inhaling, we are eating from the same, the plants being grown in the same soil, so they are, it's kind of getting transferred. So now nanoparticles are present everywhere, and that is a real threat, because we don't know what these nanoparticles, what they do once they go inside the system. We don't know the mechanism of interaction of these nanoparticle with biological systems. That's why the term biointerface, nano biointerface. So it is very important that you understand the mechanism of action of these nanoparticles in biological systems and have information what all they can do, how can they modify the biochemistry and physiology of the organism. That decision or that information is very important for any applications. Right now we are just on applications and we are not bothered because it's a very critical biosafety issue. These nanoparticles, they can cause severe effects, they can be toxic to biological systems, they can affect our vital functioning, and they can cause various diseases if they are not controlled, or if their mechanism of action or the nanoparticles which we are using for various application, if we don't do a informed study, if we are not aware of interactions, then it is very dangerous. But actually we have already reached to that point, that dangerous point, where we have now accumulation of these nanoparticles in our biological systems and in our environment. So this is a serious safety concern. And till now, whatever work was being done, people were, although this is people who are working on nanotechnology, they know it very well. Whenever you develop a nanomaterial, its property changes, whether it's chemical property, physical property, magnetic property, electrical property, even the surface area is enhanced several fold more. So the property of nanomaterial are very different from the routine material or non-nanomaterial. So we are used to, or we use this non-nanomaterial regularly in our life, and we know their effects, but we don't know the effects of nanomaterial on biological system, and that is a very serious safety concern. That's why there's a need to understand mechanism of action of these nanoparticle in biological systems. Okay, so molecular mechanism of NP interaction with biological system needs to be understood before going ahead. Now the next term which I mentioned about nanoproteomics. So nanoproteomics, again, the term itself tells you that it would somehow would be including both nano and proteomics. So when you talk of nanoproteomics, there are two things. One is nano analytical platforms, and the second thing which I'm going to describe in a bit of detail is protein corona, what we are doing in botany department at Delhi University. So nanoproteomics is integration of nanotechnology with proteomics. And this branch, it originated because there were certain limitations when we are using traditional proteomics methods to answer some questions. So a need was felt. That's why this area where you are working with smaller populations of cell, and you are using very small or miniaturized equipments to get answers. So basically, you are working at nanoscale. Your equipments you are using are very, again, at nanoscale, plus the cells which you're using, you're probably working at single cell level or group of cells. Okay, less than 5,000 cells. So that is nanoproteomics, when the scale is downgraded to nanoscale, both of techniques as well as of the material you want to analyze. Then the second thing is about development of such techniques which I mentioned to you, that these have already been developed, like you have nano electrophoresis, nano chromatography, and now we are actually analyzing single cells using these equipments. So this is one idea of nanoproteomics. The second one is about nano protein corona. I'll come to that in after a few slides. So because of so what happens when you combine both the areas, you can reap the benefits of both. Like real time multiplexing is possible because of high surface area to volume ratio of nanomaterial. Then you can use low sample concentrations to answer your question, again, because the sizes and shapes of these nanoparticles are tunable. They can be changed or controlled. So this property helps us in going for low sample concentrations. Similarly, less reagents we can use and we can answer our questions. very specific questions you can answer because of specific bindings, because these NP, their surface are functionalized. You can attach a particular group and then you can exploit property of that group to either harvest some other group binding to that, some ligand which is binding to that group, or in general to even enrich some compounds from in vivo using those binding groups. Then you can immobilize enzymes on these nanoparticle surface, like trypsin has been immobilized, and then its efficiency increases several fold. Similarly, electrical conductance of these NP is better or higher. So even that property is used in when you are miniaturizing the whole system. And even particle aggregation, because whenever these NP they aggregate, that means they become bigger in size, generally there's a color change. So even this property is being exploited to develop enzyme assays. You can have the single NPs and you add a factor which leads to aggregation of these, and then you use that property as enzyme assay. So those things are already being done. So these so what all is possible? You can do better peptide fractionation, because these NP based columns are excellent in their stability. They are very good packing materials. Then because NP surface, as I mentioned, it can be functionalized, so you can get protein enrichment, and you can even get some biomarkers which are in very low concentrations, but because of this functionalized group they are enriched. Similarly, this I've already explained, trypsin can be immobilized and its efficiency can be enhanced several fold. Similarly, interaction studies can be done, and quantum dots are a kind of carbon based NPs which are being used as labeling agents, because they give different type of fluorescence, so they can be used as labeling agent for detection of proteins in microarrays. Then nanofluidics, I think I have a slide. Nanofluidics, basically the low scale working of proteomics. So I'll just give you three examples of what I have described, why nanoproteomics has gone a step ahead to routine proteomics. So as I mentioned, you can do protein enrichment. This is one example where this is a phosphate binder. Basically, this is your iron oxide nanoparticles, which specifically binds to phosphate group. So what would happen when you use such an NP with these groups? Protein containing phosphate group would bind to them. So you can enrich phosphoproteins. And then once you identify this is your before enrichment, you see just this much peak, and this is after enrichment. The peak is enhanced several fold. So you have basically enriched your phosphoprotein. So this is one example where you can have group-based enrichment of substances in biological system. Then this is another example where what they have done, they are testing urine samples for antigen, and what they have done, they have put this urine sample with the gold nanoparticles, and what happened, these proteins from urine, they got bound to these NPs, or and then you can discard the rest of the supernatant, so you will have NPs bound with proteins, so you get an enrichment. And this enrichment, this can be seen on the gel. These are the samples where they have put NPs. So this is the initial sample. This is after processing with nanoparticles, golden nanoparticles. And these are the acetone or TCA precipitated sample. So it was equivalent to your TCA acetone precipitation. So you can enrich samples where the concentration of proteins is low. This is another application of nanoproteomics or nanoparticles. And this is just one. I'll quickly go through this. This is a system. It's basically a nanoscale system which is used for working with proteins at nanoscale, very small scale. This system is called SNAP, Simplified Nanoproteomics Platform for Reproducible Global Proteome Analysis of Nanogram Protein Quantities. So you can identify protein at nanogram level. So basically, this is a combination of right from sample loading. This is your sample loading place, and then from here there are several pumps. These pumps, basically they'll clean the sample, and they'll after removing all impurities from the sample, you have one this chamber basically contains your enzyme trypsin. So once the sample is put here, it will be cleaned, and then it is sent to this chamber where it will be trypsinized, and again it will be cleaned through the next pump, and it will be fed to your columns for separation. And eventually, after separation, it goes to MS machine. So the whole thing it works at nanoscale. So you are using less amount of sample, but you will get all the information which you need. So you use very small amount, up to 25 microliter of sample of nanoscale concentrations. So these are few examples where nanoproteomics is being used, and this is the last year's paper which was published, I think, in Nature somewhere. Yeah, Nano Letters, sorry. This is a device. It's basically a nanosensor which they have developed. This is a carbon nanotube, and to which they have attached the receptor for your corona protein, basically ACE2. By this is the receptor for protein, corona protein. So as soon as the protein comes, it will start on binding. It will start fluorescent, giving fluorescent light, and there's a quite a bit of difference in the fluorescent light on binding. This you can detect. So this is being proposed as a detection mechanism for this virus at, again, at nanoscale, 12.6 nanometer detection limit, nanomolar detection limit of this sensing protein has been analyzed and published. So this is again a very good example of a nanosensor for detection of this virus, coronavirus. So now the last part of the presentation, which where I would be also speaking about our work which we are doing in the lab. And what I've done, I have taken most of the data out from this. I just want to give you a feel of the area, what questions we are asking and what answers we are getting out of that, just to keep it simple for the students so that they can grasp what this area is about and what questions we are asking and how we are trying to answer them. Be as a tool to understand mechanism of action of NP in biological systems. So I would be telling you what is protein corona, how it is formed, what factors they affect the formation of protein corona, and how do we analyze this protein corona, and then what do we do with the results which we get from this protein corona proteins or proteome. So what we have done, basically, we have done proteome analysis of protein corona by label-free, gel-free, quantitative analysis. So this is what a protein corona is. When a nanoparticle enters into biological system, it'll face everything. It'll all the biomolecules, they will just start interacting with it. Okay, this is as it has entered into the system, and after some time, the biomolecules, it could be anything: DNA, lipids, carbohydrates, proteins, anything and everything can bind depending on the affinities and the interactions. But because we are working on proteomics, you are more, and in fact there's a kind of information available in literature that mostly proteins, they get bound more in more amounts on these NPs. So once they start binding, there are two stages. One, the initial stage, once the NP is entered, the proteins start interacting with it, it leads to attachment of some protein. Okay, this is called soft corona, the first stage formation. And after some more time, when I'm saying time, this normally take about 8 to 10 hours, and this is after that. After some time, what happens, the protein, they are getting exchanged. Okay, some protein has got bound. Now protein interaction start. The protein, the first layer of protein which have got bound, now they other proteins from the cell, they start binding to these. So protein interaction starts, and a stable corona is formed. That means a stable layer of protein on NP is formed. This is called hard corona. Okay, and this is a stable corona. This is a transient form, the soft corona, and hard corona is a final stage, or the mature corona. So NP protein corona is basically coating of proteins on the surface of NP from its surrounding as it enters the tissue or cell. And these kind of studies they have been done in good numbers in human protein corona analysis, basically for developing anti-cancer strategies, for drug targeting, for knowing the biomarkers for various diseases. So those kind of analysis have already been done in animal system, but in plant systems, you can count the number of groups which are working on protein corona on your fingers. Even the paper, published paper, if you see, you'll get most of the paper in animal or human system on protein corona. Plant protein corona, hardly any papers. Although in last 2-3 years the field has picked up. Now you see good number of publication on PubMed, but still not many papers. Those publications are mostly reviews or initial kind of studies. And I'll be mentioning that what are the problems in these kind of analysis. So it basically this protein corona analysis, it would affect your nanoparticle distribution or the protein corona formation, because now proteins get attached. So the movement of this NP in biological system would also be governed by these proteins. Even the interaction of this nanoparticle with other different molecules would be different now when the proteins are attached on NP. They would not be functioning as usual. They can acquire some new properties, or their binding affinities can change, their catalytic activity can change, their interaction with other proteins can change. So these are the various changes which are anticipated, which can happen. So, and this protein form of formation, it would depend on the type of nanoparticle you're analyzing, and also the extract which you're using, from which source you are using extract. So these are the factors which basically affect your corona formation. NP based factor includes the material which you're using. That means the type of NP you're using, its surface, what kind of size it has, what kind of charge it has, and what is the shape of the NP. All this property of nanoparticle would affect your protein corona. Similarly, protein specific, your species specific factor includes protein composition, what kind of proteins are there in the sample, then exposure time, pH, temperature. These are all the binding conditions which would change your corona. If you change these conditions, the corona formation would be affected. And these protein corona, how they have been used, some examples which are existing there in literature, they have been used for drug delivery, specifically in human, and in case of plants, nutrient delivery. Some feeble examples are there. Then ligand discovery is possible. As I mentioned, engineered nanomaterial can be used. If you hook on some binding group, you can take out that ligand. As we just now we discussed about phosphoprotein enrichment. So you can fetch out things from biological system if you can engineer this nanoparticle accordingly. You can tag a protein, and then you can get the protein binding to that protein from system. So those kind of applications are possible. Then the toxic effects of nanopollutants, they are being mitigated using, these are all proposals. They can be mitigated. Similarly, waterborne pollutants can be removed by using some engineered nanoparticles. Then biomarker discovery, I've already given you one example. And this our results are also indicating on this. So I'll be speaking on this in some of my slides. So this is the work, basically, analysis of gold nanoparticle protein corona of brassica juncta, which is Indian mustard, to generate useful information for safe and optical nanotechnology. Idea is, go ahead, design nanoparticle, design nanomaterial, but you should be aware of its effect or its interaction with biological system before using this, so that you know. So you should have informed information about the system already, and then you use it so that you know what all it can do to the biological system and even to the environment. So we are basically talking of biological system or plants. So we asked ourselves two questions, two very straight questions: comparative corona analysis with crude and nuclear extract, just to understand whether the binding of proteins on NP, and as I mentioned, we are using gold nanoparticle. So on this NP would be on basis of concentration. Say high abundant protein would bind first, and then your probably low abundant, high affinity low abundant protein would take place of this abundant protein in hard corona. This was the question. And the second question which we asked ourselves was, so this first part was done using chemically synthesized nanoparticle, and there's a reason for that, because you can such studies, the size, uniform size is very important. If you there's a change in size, your properties of NP would change. So repeatability of result would be a problem. And the second question which you asked ourselves was comparison of green and chemically synthesized gold nanoparticles, whether they will behave similarly or they'll behave differently with regard to protein corona formation. So these were the two simple questions which we asked ourselves. And For that, answering the first question, we synthesized our nanoparticles using a chemical method. Usually, your trisodium citrate is used, and at high temperature, this NP formation happens, and you are using the gold salt, chloroauric acid, tetrachloroauric acid, the gold salt which you used. And once you have synthesized this, you will see a color, say the color is towards cherry color. That is the color you get when you synthesize gold nanoparticles. And once you have synthesized them, then you have to see their size. So sizing can be done by simple UV spectroscopy. Plus, you have to characterize that they are of the same material. You have to do diffraction studies. You also can do FTR spectra just to know the working group which are sitting on the NP. And besides this, TEM is also done to know the shape of these NPs. There are various categorization or characterization tools which are available, which are generally used to characterize these NP before you use them for any application. And this is the workflow which is generally followed to understand or to extract this protein corona from a biological system. So simply you make the extract, you incubate it with NPs under certain conditions, and these conditions needs to be standardized for each extract and each NP. Basically, you are now looking for the pellet which has bound protein on it. So you have to remove anything extra, that means the free floating proteins and free floating NPs from the system by various round of centrifugation, and then eventually you just have the pellet which would have the bound protein. And that pellet, basically you go further analyzing it, you give the samples to mass for MS identification. And these are the various tools which I mentioned, which are available to analyze these proteins, like UV visible analysis. SCS can also give idea. Then MS analysis is done to identify the protein, and then these are potential to know the charges on the surface of these NPs. Then DLS sizing to know the size of these NPs. TEM again to know the surface and size. FTR to know the active group present on these NPs. And this is again a representative slide showing you the results on SDS. So this is the corona protein formed at different time points with crude sample, right from 0 hour to 36 hours. So you can see very clearly two types of peak. One is this R1, okay, and second one is this. The R1 basically is your of soft corona, and this you can see on the gel also. You can see two type of profile. This till 8 hours, proteins are getting bound, and only similar kind of protein are getting bound. They're just getting enriched. But after 8 hours, say at 16 hours, there's a switch. Here you see this is Rubisco. This just vanishes. Very small amount is there. In fact, other proteins start binding. So there's a change in the type of protein which are binding from this soft corona to hard corona. And after 36 hours, this stabilizes, because you see almost similar profile in all at different time points. So with your crude samples, you could see two different type of corona, soft and hard, getting formed. While with your nuclear extract definitely it would be relatively a cleaner extract in the sense, because only nuclear protein would have limited functions. Whatever the nuclear protein functions they have to perform, only those would be there. But in crude protein, you will have all different type of proteins. In fact, diversity of protein would be high. Here, relatively, they won't be that diverse, and that we can very clearly see on corona formation. The type of curves which we get, they are almost similar. You don't get biphasic curve like you get in crude. And this you can see here also. There's just simple enrichment of proteins. It starts from here, and the profile is almost same till 36 hours. So that is one clear difference. At least binding wise, we could see that in case of nuclear proteins, the binding is kind of uniform, and only one type of corona is getting formed, or one type of peaks we get. Okay, and this is the additional characterization where we see your TEM to know the size. And so initial, this is the particle size of average particle size which we used for the experimentation. This was 21.5 nanometer, and this is after the corona protein corona is formed. It almost became double, slightly more than double, 55.75 nanometer after binding the protein layer. So this is a protein layer which you can see here in this picture. And so when we did this UV scanning of these, we could see, as I mentioned, two different regimes in crude fraction, R1 and R2, while in nuclear it was the same. And so that was the difference. The question which we asked, whether we will get different type of corona getting formed, it was experimentally proved that yes, if you change the extract, you get different type of corona getting formed. So the type of protein which will bind and the way they would bind would be different. And this is a slide on this analysis when we did a bit of analysis of these targets. So we basically get thousands of protein getting bound to these NPs. I'm not giving you the list or the numbers, but these broad analysis which we do with this two samples, what we could see that when you, this is basically your light bar is soft corona and dark bar is your hard corona or stabilized corona. Again, the same kind of information you see even these pictures, different sizes of proteins in crude, right from 16 kDa to 60 kDa. They are binding to the NP surface, gold NP surface in case of crude extract, while in case of nuclear extract, it's just 10 to 20 kDa protein, which are binding in higher number. So molecular weight preferences are slightly different, and they are broader for crude. Again, the reason would be diversity is more here, so you have a broader molecular weight preference in case of crude in comparison to nuclear samples. Okay, and this is again a bit of analysis on I think it was about pi, your charges on the protein corona. With crude proteins there was a shift. Initially there was a high pi groups which are binding in soft corona. This is again a comparison of soft is the lighter bars and dark bars are your hard corona. So initially you see, if you see the soft bars or the light colored bars, you have one group is this and the second group is this. Negative charged, this is one class, and positively charged, this is a second class. So high pi or positively charged proteins were present more in crude corona initially in soft corona, and later on it's changed to other kind of charge. But in nuclear it was almost uniform, hard and soft, they show same kind of binding, like there's no preference or no such distinction with the nuclear proteins. And this is just on the localization, cellular localization. If we compare the binding, which type of protein from which location of the cell, what from which compartment they are binding more. So if you see again, it's a comparison of crude corona and nuclear corona. Crude corona is basically your crude extract corona, dark one, and the soft one is lighter one is your nuclear corona. So in soft corona, you have more of, in nuclear corona, you have more of nuclear protein, which is expected, and ribosomal proteins are also there. And in crude corona, lesser of nuclear protein and good amount of your ribosomal protein. But if you see here in hard corona, nuclear is still maintaining, the nuclear protein are still bound. The crude one, slight increases there, and ribosomal, because ribosomal are also kind of nuclear protein because they are present in nucleus, but eventually they are used in cytoplasm. So initially the assembly and all happens in nucleus. So there were clear distinctions in the localized or location-wise binding of proteins also. So first question which we could answer was that the corona formed, or the type of proteins which are sitting on the corona in the two extracts are different, and even their affinities are different. Okay. And this is just, we ask the next question, whether these proteins which are sitting on these NPs, whether they still maintain their activity or not, or they lose their activity. So we checked for 2-3 enzymes, malate dehydrogenase, fructose bisphosphate aldolase. These were the targets which were present on these, and they were checked for their activity. Again, this is your the light one. This is total leaf soft corona. This is total leaf hard corona. And this is the dark one is fructose bisphosphate aldolase, and the light one is malate dehydrogenase. So this is in crude sample. Then in this is in soft corona and crude hard corona. So initially, when you see in soft corona, you see some activity, and in later this activity decreased a bit, but the activity in hard corona is equivalent to your crude sample. So activity is kind of being preserved or maintained. We also asked another question because, as I mentioned, Rubisco was coming as a target. So whether what happens to Rubisco on binding on these NPs. So we did CD analysis, and what we could see that beta sheet dominated structure got enhanced on binding to NP. This black line is simply the purified Rubisco, and this is on binding to NP. So beta sheet structure getting enhanced means it's stabilizing Rubisco. So that was a good point. That means on binding to NP, Rubisco is stabilized. So that means it might be translated to higher efficiency of Rubisco. So that we still are on that. So this is where this part ends. The second question which you asked was whether the binding affinities of chemically synthesized and green synthesized NPs would be different. And we didn't find much difference as far as binding is concerned. We almost saw similar kind of curves in both, and even the gel profiling was almost similar, not much differences. But when we investigated further the proteins which are bound, so green hard there were about 1319 protein which were identified, chemical 1268 proteins, and this is in hard and this is in soft. So initially you see fewer proteins are binding, and in hard, once the it is stabilized, higher number of proteins are getting bound to these NPs, both soft as well as green. But few distinctions we could see, like in your green one, this is the green one, the category of protein which are binding are varied or diverse in green. In chemical, relatively fewer type of proteins, that means category wise or function wise, fewer category functional categories of proteins were binding. In green, diverse category functional categories were binding, and this probably could be explained because of the varied charges or varied groups which would be present on green synthesized nanoparticle, because they are using inherent compound for synthesis. And once we compared all the data, what we could see that the soft corona was almost similar in both green and green and chemically synthesized NP, but the hard corona was having more proteins. This is or the positive side, that means higher number of, actually higher quantities of protein which are bound on chemical, but in green the quantity was relatively less. I'm mentioning two things, quality as well as quantity of protein. When you talk of quality of protein, you get better quality or more variation in quality when you see the protein sitting on green NP. But when you see amount or quantity of protein, you get more protein bound on chemically synthesized NP. Okay, so this was a very clear relationship which we obtained after doing this PC analysis, and this results also corroborate the same. This is a comparison of, basically this is a MapMan metabolic pathway which we have drawn using all these proteins, and you can see mostly red boxes here, all throughout. These are all more of red boxes in comparison to blue one. Blue is your green hard corona, and red is your chemical hard corona. So as I mentioned earlier, more amount of protein, quantitatively more amount is binding to chemical hard corona of chemically synthesized NP, and all these participate in different pathways. They were put on this pathway map, and we could see that the chemically synthesized or hard corona had more amount of protein, and they belong to different pathways once MapMan metabolic pathway analysis were done. So I think last few slides, just to give you an overview, what all we know about protein corona in plant. They are just these significant 4-5 studies which are available, where in one case they have done analysis with xylem sap, and what they have seen that when you see analyze this xylem sap, more proteins are sitting on biocorona. They call it biocorona. Biocorona because they are trying to analyze all different components sitting on the, or different macromolecules sitting on the NP. So, but they found more of proteins. So it was majorly a protein corona, not a biocorona. And normally, this I've already explained that beta sheet content increases of Rubisco, and this we have published recently. Then we can concentrate biomolecules, and this is one example where they have done nano harvesting of metabolites. This paper was published in Nanobiotechnology in 2020. Then this is another paper where they have analyzed the rhizosphere, because this corona formation, once the NP are going, they are going through root. So they'll be interacting with all the surrounding microorganisms and chemicals which are present in rhizosphere. So even there they would be modified. So these people have done those studies, and they have studied the NP uptake, and they have done the phytoenzymes which are excluded in ribospheres. How do they affect these carbon-based NPs? And this is another interesting study which is published in 2020, where they have done nano priming of seeds. They have used manganese nanoparticle, and what in this particular study they could see formation of nano corona. They used a dye, and they could show presence of these protein corona inside the seed. And this is the first example of showing in vivo existence of protein corona. So a very elaborate, or I think I'll come to this point in the next slide. So this is a summary of what work has been done regarding protein corona, and these are the major issues or major bottlenecks. Why there are fewer groups which are working, why there are fewer paper on this research area on protein corona research. One, controlling the parameters which leads to corona formation are many, and it's difficult. There are various parameters which control this corona formation. So controlling those conditions and optimizing the procedure is a tedious job. It takes a long time, and you have to control various parameters. Secondly, the second point is that as far as plant corona, protein corona research is concerned, in case of animals, it is now well established, especially in human, that when you compare the in vivo corona and in vitro corona, what is the meaning of that? In vitro means when you are putting the extract and NP together in a test tube. That is in vitro. And in vivo is when you are looking at the protein corona formed inside the biological system. You are extracting that and analyzing that. So that is not possible for plants till today. Those studies are not available. So we still don't know how different the in vitro and in vivo corona would be. So whatever in vitro studies are being done, how much of it would be applicable or would apply or extend to in vivo protein corona? This is another big question which is still to be answered for plant protein corona studies or analysis. Third, the equipment which are required for analysis of this protein corona are very sophisticated, and generally they are not present in plant departments. Okay, so this is another big limitations. You have to collaborate with chemistry people or physics people. And then lastly, the workforce, because it's a new area, so there are very few people who are trained in this. So we have meager workforce for this area of research. Then the major conclusion of whatever I've presented till now, I think I'll finish in another 2-3 minutes. These are last few slides. I can just mention you, I think, some of the points. I'll skip this because I think I have already spoken about some things. Like I've mentioned about Rubisco. Even besides Rubisco, there's another important point, carbonic anhydrase. This enzyme is also important in your photosynthesis mechanisms. So right now we are trying to correlate, we are doing in field experiments where we are trying to give this NPs, and we are trying to relate these Rubisco activity and carbonic anhydrase activity with photosynthetic carbon capture efficiency. And this work is still going on. So probably we'll be able to correlate the things like photosynthetic efficiency and the stabilization of Rubisco and carbonic anhydrase on corona would be correlated. So this work is still going on. And this is one big, I have already mentioned this, that in vivo characterization of protein corona is still awaited in plants. Plus the biocorona, like the other things, lipids and carbohydrates and other things which could be present on this corona, still needs to be investigated. And this is a publication which appeared in 2020. And I think these are a few 2-3 important points. This I've already mentioned, that we are trying to correlate it with photosynthetic yield. Then in vivo protein corona is another important point, and this is another important point which needs to be mentioned. Like whenever these NP they enter the plant system, they enter through your cell wall or cuticle or roots, but they have to cross your apoplast and they go inside. Wherever they go, say they go to nucleus, but they have to come from apoplast, then they go to cytosol, and then they'll go to the organelle. So at each stage, the corona would be different. As initial experiments have shown, as the extract changes, the corona also changes. So ideally, your protein corona would vary at each step. So those sequential interactions or sequential study needs to be done. So that is very important to understand what the final corona would be. Okay, even transportation, how it would be, and how much is going to which part, those kind of studies. So actually, it's the beginning of this area. Lot needs to be done. Lot needs to be studied. And this is the group. I think few years back. Now many of the people have left the lab. Some of them are there. Like this is Ankita. She is in some DU college. She's teaching as a doc. Bhavna, she's in KMC, I think. She has, Minakshi, she has submitted her thesis recently. She's waiting for her Viva to be done. And this is Satya Prakash. Basically, this is his work majorly. He's working on this area. This boy is in Hungary. I think he did MPhil from our lab. And this is Jasmeet. She's in Australia now. She was the very first batch of students. Shruti has submitted her thesis, but she's still continuing as a postdoc in the lab. Priyanka is about to submit probably in another 6-8 months. Yai is ready to submit. She's writing her thesis. And this girl, she has left our lab. She's in IP now. So this is the group. And we also conduct proteomics workshop, as Ruchi mentioned. So this was the first one, which was covered by Proteomics Journal. And this is just a glimpse of these workshops. So these are very engaging times and very interesting times where we teach proteomics to people so that more and more people are trained in this area. Thank you for your time and for your kind attention. And the work was supported by DST and DST Purse Grant and Delhi University R&D grant. I think that's all from me, Ruchi. Now you can take over.
Thank you, ma'am. Thank you very much. Ma'am, kindly try if you can switch on your camera now.
Okay, let me try. Just give me a minute. Huh?
Right, ma'am.
I'll escape this presentation, and I'll probably try to rejoin again if
In the meantime, Dr. Ravi Gupta, ma'am, your student has also joined for your lecture.
good,
And
good. That's good.
our next resource person means
Mm-hmm.
Pratima and Shubhangi Kalla. They have also joined for your lecture.
Good. Yeah.
And ma'am, actually, thank you for delivering such a comprehensive and such an illustrated talk. And you have started with such a simpler way. Means you started with the central dogma, and then you explained very basic terms, and then you moved on to nanotechnology, proteomics, and then NP protein corona. Ma'am, there were several terms which were new to me as well, but I'm sure that students, they have learned a lot from this lecture, because as I mentioned that you have started with central dogma that our student actually know. And ma'am, Really, it was such an engaging lecture with such beautiful slide. Ma'am, if you permit, I just want to ask several questions that are
Sure,
being
please.
asked by Telegram group by the participants. So,
Yeah.
ma'am, the first question is, in between gel-based and gel-free method, which is the
Mm-hmm.
most suitable and economical?
Actually, economy wise, if you talk, gel based is more economical, but suitable, this depends on what question you are asking yourself. Sometimes when you are analyzing, say, PTMs and all, your sample size is small, and you have to resolve, you have certain procedures by which you can enrich those PTMs. So in those cases, even gel based is suitable. But if you want to ask a question like we are asking, what all proteins are binding on these NPs, then you have to have a broader coverage. So there you need a lot of information. So there your gel free system, they are more appropriate.
All right, ma'am. Thank you. Um, then, ma'am, second question. Can the proteomics analysis be done at community level for microorganisms? Again, I'm repeating question, ma'am.
Yeah, just
I
repeat it.
don't
Now
think
I
you
can
can.
see.
मैम
Now I
आपने
can.
दो डिवाइस से ज्वाइन किया है क्या?
हाँ जी.
मैम आपने दो डिवाइस से ज्वाइन किया है?
Actually, sorry. My camera is working on this one. It's the same device, but I've joined again.
Okay,
So,
now because मैम आप एक साइड का माइक ऑफ कर दीजिए because voice is echoing.
अच्छा, I'll just do that. But ये तो off ही है इसमें तो. On one it is already off.
Ma'am, you have to mute.
No, the mic is on. The mic is on. The mic is on. The
मैम
mic
see
is on. The
आपका भी
mic
वॉइस
is on.
इको कर रहा है.
Hello. Is it still echoing? Is it still echoing? Is it still echoing? Is it still echoing? Is it still echoing?
Yes, ma'am.
It is.
Just a moment. I'll try to solve it myself. Ma'am, please, now I think it's okay.
I think ma'am is still sharing her screen, so she can stop sharing.
All right, ma'am, no worries. Can you hear me, ma'am?
Ruchi?
Ma'am? Yes, ma'am. I think there is some problem with the
Okay.
camera. Ma'am, no worries. I should ask you the questions and
चलो ठीक है. No issues.
okay,
Let's finish it off. हाँ.
right, ma'am. Can
हाँ.
the proteomics analysis be done? Oh, ma'am, it has started working now. Ma'am,
Oh.
kindly adjust the camera a little bit. Maybe you can move your
Because I
device.
cannot, actually I cannot see it. Okay, like I can see it now. Yeah, yeah.
Yes, ma'am.
But there are
Now your
windows on my
face is
back.
no problem, ma'am. No
अच्छा.
problem.
हाँ. हाँ
So
जी
happy to
बताइए.
see you, ma'am. And
हाँ.
I'm thanking you again because बिल्कुल पता चल रहा है that you are not keeping well, पर from your busy schedule, ma'am, you have taken out time for us. Thank you very much again,
No, no, no. It was a pleasure. It's always a pleasure. So,
ma'am. Can the proteomics analysis be done at community level for microorganism?
for microorganism at community level, because I generally don't work with microorganism, but I think it should be possible. That is what I can tell you.
Okay. Then
Because if you have genome information, you can always dig out information. Eventually, you have to match. Okay.
all,
So if
thank you.
genomes are available, then you can always do it.
All right. Do nanomaterials cannot be toxic to environment and cause contamination?
Sorry, can you just repeat your question?
Yes, yes. Do nanomaterials cannot be toxic to environment and cause contamination?
You're saying two nano engineered material.
Ma'am, the question meant that maybe nanomaterials are toxic to environment and can they cause contamination? Nanomaterial.
Definitely, they would cause. Definitely, they would cause contamination, and they could be toxic. If higher amounts of these nanomaterial they are accumulating, they would definitely be toxic.
And their size, as you mentioned, is a big problem, that they are really
Yeah.
very small.
Yeah.
Right.
Yeah.
Thank you, ma'am. Then how is nanotechnology associated with proteomics?
I've already explained there are two levels when you
Right.
talk of nanoproteomics. One is you are working at miniaturized scale, at nano scale, and nanoprotein concentrations. And you are using specialized designed equipments for that, say nano columns and nano devices, say SNAP one I described, where they are using a miniaturized pumps and all for analysis, proteome analysis. And second, which I mentioned was about protein corona, which is actually you are actually using nanomaterial and you are trying to analyze interaction of proteins with those nanomaterials, and the major aim being you want to understand the biointeractions. What happens once the NP goes inside the biological system, how it interacts with proteins, how it would modify the biochemistry and eventually the physiology of the organism. So those kind of studies, they also come under nanoproteomics. The protein corona analysis is also a kind of nanoproteomics.
All
Fine.
right. Thank you, ma'am. Actually, ma'am, most of our attendees, as you know, today is GBM in the botany department. And secondly,
Yeah.
second and final year students are having their final exams, right? So, ma'am, most
Okay.
of our attendees, they are first year students and some faculty
Oh,
members from other universities. So maybe, ma'am, because
okay.
this is something very new for them. It is new for me as well. Some
That's why I
terms.
tried to keep it very
Right,
simple.
right, ma'am, right.
Yeah.
Ma'am, the last question,
Mm.
how feasible is the use of protein corona? I'm repeating again, how feasible is the use of protein corona or nanoparticle in agriculture when applied in field conditions? Should I
Yes.
repeat, ma'am?
No. It's all right. Actually, it's basically translation of this information. That is the question. How this information can be translated to field. That is the question. So the
Right.
precise reason of doing this analysis is to understand the interaction, so that you beforehand you know what kind of interactions. And as I mentioned, this is just the beginning. You have to do these analysis for different types of NPs, for different organisms under different conditions. It's just the beginning. It's just the tip of the iceberg, I'll say. A lot needs to be done, and once you understand the interactions properly, then if you use the nanomaterial, you can design them such in such a way that you can precisely apply them for whatever you want to do for. So the tight control would only come once you have done all the analysis and understanding what groups to put, what do you want those groups to do. So once you know the biology of it or understanding of the mechanism of it, then you can control it proper. Right now what is happening, people are using NPs just like that. They are using NPs. I am not criticizing. A lot of applications are there, they're already reported, And a lot of things are happening in even in agriculture, nanotechnology, agro nanotechnology, like nano urea, nano nutrients, nano pesticides. They are there and they are being used. But point is, what happens once you apply them? What else besides this benefit? What negative effects they are having? So to understand those things, it is very important that you understand the biology of it first. Once you understand the biology, then you can control them well. That's why the term informed usage. You use, but you use with the information, and you using them in a precise manner, so that you just do what you are desiring to do, and undesirable effects are kind of taken care of. That is the purpose of these whole protein corona analysis. And definitely in future I envisage a lot of application, but in an informed way. That is the purpose of these studies.
All right, ma'am. Maybe if we are not doing it in a informed way, we will be adding to some sort of
Yeah,
pollution
yeah, yeah,
in
yeah.
agriculture. Like we started
we
with
are
DDT
spoiling
and
we are spoiling our
yes,
environment, we are spoiling our biology, we are spoiling, or we are negatively impacting all life form, even rhizosphere and all. We are kind of damaging it if it is not done in a proper way.
correct. Thank you very much, ma'am. Thank you very much. And if anyone wants to, any of our participants or esteemed speaker also wants to interact with ma'am, you can raise your hands. Uh, if anyone wants to ask any question. All right. Thank you very much, ma'am, for sparing your valuable time. It was really great to having you here. The person who has actually started proteomics in the botany department. Thank you very much, ma'am.
It was a pleasure. It was a pleasure, Ruchi, and it's always very nice to interact with students and to share information with them, so that they are kind of
Yes, ma'am.
excited and they know about the newer areas
And
and they
ma'am,
know.
I am one of your first students, MSc previous
I know
in the
that. I know
year
that.
I think 2001.
That
Ma'am,
is the time when I joined the department.
yes, ma'am,
Yeah,
you joined the
yeah,
department in
yeah.
2001. Ma'am, it was great having you here. Thank
No,
you very much again, ma'am.
it's nice to see all of you here, and it was my
Yes, ma'am.
pleasure.
We have Dr.
And
Zishan also. Maybe
okay,
you have taught him also. No, maybe he
Jishan is
has
also
joined
from
you.
botany. Is he from
Yes,
botany?
ma'am. No, ma'am.
Oh,
He has done his post graduation from Allahabad, and
acha,
he is Dr. VP Singh sir's student,
acha.
and he's teaching in our college since 7 years.
Okay, okay. So probably I've seen him because face look familiar, but वैसे एकदम जान नहीं पा रहे हैं कि
Right, ma'am.
फेस तो
Ma'am,
इनका familiar लग रहा है. So
now you plan a proteomics workshop. It will be great learning from you in the lab, offline.
definitely. अभी तो
Yes,
कोरोना से हम 1-2 साल तो कर नहीं पाए ना, so we had a
yes, ma'am.
online
There is a
conference
gap.
only last year,
Correct.
but
Ma'am,
probably
let's,
because next year
right.
it will be 10 years of these workshops, so probably we will have something
Yes,
nice. I'll
correct.
think about that. And it is always a pleasure to have you people and interact with you people, and seeing you
Offline, ma'am.
flourishing in different colleges and all. So it's always
Correct.
nice to see you. So thanks a lot, Ruchi, for this opportunity, and
Thank you very
congratulations to all the organizing committee members once again. And I think this is going well. So I think I'll take off now because, as we have a GBM, so I have to
right,
just
ma'am.
brush up
This is
myself for
what
that.
I want to mention, that today is such an important day. You have GBM in the department, and then also you have joined. Thank you very much, ma'am. Thank you.
Thank you, Ruchi. I'll just call off myself now. Thank you.
Now over to you, Dr. Zishan. I think Pratima is there, our next hands-on speaker. Dr. Pratima, Miss Pratima, you can switch on your camera, please.
Thank you, Dr. Ruchi. Am I audible?
Yes, yes, Dr. Zishan.
Thank you. Uh, Good morning, uh, speakers, resource persons, and dear participants. Now I take this opportunity to invite Ms. Pratim Verma for the hands-on session. Good morning, Doctor. Good morning, Ms. Pratima.
A very good morning to all of you, and thank you for inviting me in such an important workshop. I'm very delighted to be part of this workshop, and thank you, Ruchi ma'am, for inviting me. And over to you.
Thank you. Now I take this opportunity to introduce Ms. Pratima. Ms. Pratima Verma is a PhD scholar at Department of Botany, University of Delhi, under the guidance of Professor Suman Lakhanpal since 2019. She completed her bachelor's in botany from Shivaji College, University of Delhi in 2017 and master's in botany from Department of Botany, University of Delhi in 2019. She started working on DNA methylation in plants as part of her MSc dissertation with Professor Suman Lakhanpal and continued to work on DNA methylation changes upon phytoplasma infection in sesame plants as course of the PhD, along with dual transcription and effector biology to understand the phytoplasma interactions. The main focus of her PhD research is to understand the developmental alternations caused by phytoplasma in susceptible plants. Now I request Dr. Ruchi to play the video of hands-on session. Thank you.
And then we'll design primers.
Dr. Zishan, I think my screen is visible, right?
Yes,
Good
screen is
morning,
visible.
everyone.
You
I'm
can
Anjali
select the
Zakir Hussain,
order.
Delhi
Yeah, it is in
College,
full screen mode
for
and
inviting
audio is
me to
also.
talk about methylomics, which is
Okay.
an emerging field of gene expression alterations. So today I'll be talking about epigenetics and more specifically focus on DNA methylation and how to measure it. For hands-on session, we'll be analyzing whole genome bisulfite sequencing data using MethylKit. And then we'll design primers using Myth Primer and then quantify the data using QMA softwares. So this is me. I'm a PhD scholar at Department of Botany, University of Delhi. I work on sesame plants infected with phytoplasma using different approaches such as DNA methylation and RNA-seq. So what is epigenetics? Epigenetics refers to a stable heritable trait which is not explained by a change in DNA sequence. Epi means on top
Sorry
of
to interrupt.
genetics.
I think
So
the
it
video.
adds an
Okay,
extra
sorry.
layer of information above DNA sequence.
Sorry.
Epigenetics is essential for normal development. It can be modified by environment and can be disrupted in diseases. Today, I'm only focusing on DNA methylation. So now some of you must be wondering what is DNA methylation. When you look at the ATGC base pairs, DNA methylation is addition of a methyl group on the base cytosines. These cytosines are found in dinucleotides with CG, and these CGs are not these are not evenly spaced and found to be clustered in the genome and called as CPG islands. Methylation is important as it can regulate gene expression. What you see here in the plot is a relationship between DNA methylation and gene expression. As you can see, when we increase the DNA methylation, the expression of the gene decreases and vice versa. Whatever you see here can be explained by image on the left hand side. You can see there is a normal cell in which the gene body is methylated. but not the promoter regions, and this gene is getting expressed. However, in a cancer cell, for example, the methylation is present in the promoter regions, but not in the gene body. So the expression of this gene is suppressed. So there are defined roles of methylation in plants. One such is the developmental trigger of fruit ripening in tomato, which is found in the CNR mutants. These mutants are found to be methylated in the promoter regions, which suppresses its expression. When 5-azacitidine was applied to the wild-type tomato to repress the methylation, same effects were observed. So this is a model which tells us about the role of DNA methylation in the resistance of plant against the pathogenic Pseudomonas syringae. As you can see, the symptoms are observed in the wild-type leaves, but not in the MET1 and DTC mutants upon Pseudomonas syringae infection. These MET1 and DDC mutants are methylation mutants. The methylation is not found in the promoter or the TE regions, causing its overexpression of the resistance genes. And upon infection, these mutants do not show symptoms. So this is an algorithm for choosing a suitable method for DNA methylation analysis based on whether you know your candidate genes or not. If you don't know your candidate genes, you can go for whole genome methylation profiling, or you can search for differentially methylated regions. And if you know your candidate gene, you can go for digestion-based assay followed by PCR or qPCR, or you can directly go for bisulfite sequencing followed by pyrosequencing or high-resolution melting. So what is bisulfite conversion? Bisulfite conversion is a chemical process of converting unmethylated Cs to Ts or the uracils, while the methylated Cs are protected. These create SNP condition, which are used to call methylation. As you can see, this is an unmethylated C, which is converted to uracil. However, the methylated C is protected after bisulfite conversion, and this can be detected in PCR in further analysis. So now that you know methylation is cool, but how do we actually want to know about it? So there are two types of bisulfite sequencing: whole genome bisulfite sequencing, which is known as the gold standard of DNA methylation. It is expensive but covers almost everything. However, a targeted bisulfite sequences only targets the region of interest. It is cheaper, but we can miss a lot of information, and it usually requires a higher coverage. This is a workflow of whole genome bisulfite sequencing. It is a unsaid rule when you are working with DNA methylation. The quality of your DNA should be very much high so that you do not miss information. Once you have extracted high quality genomic DNA, you go for bisulfite conversion using sodium bisulfite, which is available in commercially available forms. After bisulfite conversion, what you do is fragment your DNA. After fragmenting your DNA, you do random priming, tagging, and PCR amplification using suitable primers after indexing or barcoding your samples. This creates your adapter tagged epigenome library. After your library is ready, you go for sequencing on platform like Illumina. Once the sequencing is done, you get your data in form of FASTQ. After you get your FASTQ files, you go for quality check using FastQC. The adapters are trimmed using Trimomatic and reads are aligned to the genome. After removing all the PCR bias duplicates, you go for methylation calling. This is the pre-processing part. Once you get your methylation calling files, you go for downstream analysis. In downstream analysis, what you do is normalization, imputation, dropouts. The sequences that are not required or you think are not suitable for analysis can be removed in downstream analysis. After all that is done, you get is your final differential methylation data files, which can be annotated and further analyzed. This all downstream analysis can be done using MethylKit, which is an R package. So let's start with analyzing whole genome bisulfite methylation data with R. So before we start, make sure that you have installed R, RStudio and RTools. And after installing these, you need to load MethylKit package in RStudio. This is a flowchart of possible operations by MethylKit. You can input a aligned file in the MethylKit, or you can go for a methylation call files. After you have input your files in the MethylKit package, you can go for downstream analysis such as uh, coverage statistics or methylation statistics. You can go for correlation, you can go for clustering and PCA and a lot more. All this can be done using MethylKit. So this is a methylation calling. You align your reads to a reference or a reference genome and see how many C's or T's are converted in the reads. As you can see in this position, out of 10 C's, 2 are converted to T's. So we say there is a 80% methylation at this position. However, only two reads showed conversion. So we say there is a 20% methylation at this point, at this position of C. So it is an unsaid rule that you need a good coverage for reliable methylation calling. So this is a typical methylation call file, which shows your chromosomal base, your chromosome number, the base, the strand, forward or the reverse, the coverage of the reads, the frequency of C's and the P's. So you input these files in the MethylKits. So we'll start with the hands-on session on handling this data using MethylKit. Let's quickly start with the hands-on session on MethylKit. So we'll open RStudio. This is my R script that I have already written. Otherwise, it would have taken a lot of time to type. For convenience, I have already written my codes that I'll be running in RStudio. This is RStudio. Already I have loaded the packages required to run MethylKit, like Genomation and one or two software that were required for MethylKit. So let me clean this. This is my R console. This is my environment, and all the plots that will be created will be shown here. Let it load. So some of the packages were already present in the MethylKit, but some of them had to be installed individually. So this all information can be found out on the bioconductor MethylKit website, and all the codes and everything is available. So there's no need to worry that you'll not be able to do it on your own. A lot of help available online. So I'll clear all this. Now I will start with the data analysis. So I'll open my library. methyl kit. So it is saying what was telling me something about replacing of previous version of genomation. So now we have opened the methylkit library. How we load our data? I have two test files or the infected files. For example, in my case, I have a healthy plant and I have a infected plant. So I'll say the infected plant as test samples and my healthy plant as control samples. Generally, the DNA methylation data is like this. You have a control sample and you have a treatment sample. So now I have 4 files that I'll be loading on R. So this has created a file list. You can see my file lists. There are 4 samples combined in one. So this is file list 1. This file list 2, file list 3 and 4. 2 test and 2 control. So again, after loading my data, what I will do is I'll read my files to a methyl raw list, which methylkit will read. Only loading my data on R will not work. I will have to feed it in a format that methylkit will understand. So what I have done is I've created my object file that will be read by methylkit. So it has read my 4 files and created it. You can see after loading the files, it will not directly be read by methylkit. So you have to create an object. And this object has created a single file that contains 4 samples. It has a list of 1900 entries with 7 columns and so on. All of them are same except that these 2 samples are test samples and these 2 are control samples. So now you have viewed your my object list. So this data is ready for analysis. So first of all, we'll apply or we will see a descriptive statistics on only one of my list. So I am doing the descriptive statistics on only the first object. So you can see this is a summary of the descriptive statistics. So all these codes, before running all these codes, you should know some of the basic working of R and RStudio. Otherwise, it's very easy to do. You just need to run the commands. So now you can see there is a summary that has been created by methylkit. And this is a methylation statistics per base. So I have only performed the methylation statistics of the first list. Similarly, what you can do is you can just change it to my object 2 and find out the descriptive statistic of the my second list. Similarly, the third and the fourth. So you can see it is telling me about the median value, the mean value, and the third quartiles. There are percentiles, how much of my entries are falling in 30% methylations, 40% methylation, 50%, 60%. As you know, in the starting, I told you that we are feeding the input base call files in the methyl kit, which had the frequencies of C's and T's. And after loading the methylation call files, these are methylation call files that we have loaded in methyl kit. We can check how much of our reads are falling in which percentile. So we can say 18.18% of my reads are falling in 40% methyl, 40% of methylation. So you have performed a methylation statistics on only one file. Similarly, you can do for rest, I told you. Now you can plot your methylation statistic using this function, which is get methylation statistics, in which you'll say if it's true, if it's everything is working fine, you can plot my data. So it is running. You can see there is a histogram created of the methylation statistics. So which is telling us that how much percentage of my reads or how much frequency of the reads are falling in 0 to 20% of methylation per base, and similarly in the 20 to 40%, 40 to 60, and so on. So this is a histogram of percentage methylation per base. It is very easy to create plots using methylkit, and not only methylkit, such softwares are available. So now you're going to plot your read coverage per base. You need to see how much of your coverage per base is done. So this is a histogram of read coverage per base, which is telling us about the frequency of the reads and log 10 of read coverage per base. So actually here we are taking the log 10. Otherwise, it would be very difficult to show it graphically because the range of the data varies. So here it is shown in log 10 values. This is a coverage per base graph, which is created using methylkit. It is only of test 1 sample. Similarly, you can do it for rest of the samples. Now that you have plotted your methylation statistics and read coverage per base, now you can also filter sample based on read coverage. As I told you, it is very important. The coverage of your reads is higher to be very much true about the methylation state of a base. If you feel that the coverage of my data is not sufficient to support my hypothesis, or what you say, is not confident. If you're not confident about your coverage, you can filter your samples based on read coverage. So the lowest count that will be taken would be 10 for filtering of your read coverage. And it will create a list of a filter data. So this is your filtered my object. First it loaded the samples, then it created a list of my objects. Now we have created a list of filtered my objects. This is the same list, but we have filtered our data. As you can see, there's a change in my object and my object filter data. The rates are less in filter data. So now that you have filtered your data based on coverage, you can merge your samples using unite function of methylkit. Now you are creating a merge file of your all samples, test and control. So it has created a myth file that have all the observation of your test and control. The same base across four samples. This is the coverage 1, this is the coverage 2, coverage 3 and coverage 4. This is the same way showing the coverage in sample 1, sample 2, 3 and 4. So you have merged your data. So after merging your data, you will create a methyl base object where only one CPG covered with at least one sample per group will be returned. There were two groups defined by the treatment vector. This is the treatment vector. I'll show you. This is the treatment vector in which you told methylkit that my first two files are my test samples and second two files are my control samples. So it will create two groups. That this has created a file called myth.min. We had taken a minimum percentage. So only the bases that were more than a minimum cutoff are only retained and rest were removed from the sample, from the list. As you can see, this one only remained in second sample and the fourth sample. It was removed from first and the third one. So now you have created two groups based on CPG covered with at least one sample per group. The CPGs that have at least one sample will be remained. There might be some CPGs that were not present, so they will be removed from the list. So to get the correlation analysis, we'll use the get correlation function. So this is the correlation between four samples, test and the control. It is also creating a plot of the same. This is our correlation plot between test and the control samples. You can save these graphs for further use using save as image or save as PDF. Save this as image. So I have saved this on desktop. do it again. This is my correlation plot. So get back to R. So now we have seen the correlation. You can also cluster your samples and plot it. So it is telling me that my control samples are lying together and my test samples are lying together as expected. So to get the real differential methylation, we use my function of methyl kit to get or to calculate the differential methylation. So it has detected two groups, treatment 1. It is saying group 1 to treatment and group 0 to control. So here, my diff, it is the file my diff. It is telling me about the position, the chromosome, the strand, plus strand or the minus strand, the p values and the q values, and the difference between my treatment and control. Minus indicates hypomethylation and the positive tells me about the hypermethylation condition. So you can also filter your data based on hyper or hypomethylation, and you can also choose only those bases that have percentage methylation of more than 25%. So now you are getting only the hypermethylated bases that have a percentage methylation of more than 25%. So this is a list of differential methylated bases that are hypermethylated as well as have a percentage methylation of more than 25%. Similarly, you can do it for hypomethylation. You can sort your data based on hyper or hypomethylation, or you can get a common list of all the hypo and hypermethylated bases. So I'll do it for hypo and hypermethylated bases together. This is a list contains both hyper and hypomethylated conditions. So finally, we'll annotate our data using Genomation. You need a reference file, which is in the that you will feed in the system. You can get your annotation file on publicly available databases. We'll feed the annotation file. You need a Genomation package for annotation. So it will tell me about introns, exons, transcription start sites, promoters, etc. So we have loaded our annotation file. So we will annotate differentially methylated CpGs. That will tell us about promoter, exon, introns. So here is the summary of the target set annotation with genic parts. This is the percent of target features overlapping with annotations. Like 27.82% has been overlapped with the promoters, while 15% to the exonic regions, 34% to the introns, and 57% to the intergenic regions. We can use this function as well for annotation purpose only. Differential annotation by gene parts. This is the same thing that is telling me about the annotation percentages in different regions. The same thing in different formats. You can also find the association with the transcription start site. In this list, you can see we are also getting a, but it is only showing me the 6 values. It is not showing me the whole list. We have used the head function, so it is only showing me 6 values. Also, it is showing me the feature name. Like if you can just copy this and search on NCBI for the identity of your genes. You can do a blast search to know which gene is corresponding to which region of methylation. You just don't need to do this. It's just extra. Now that you know the identity of genes that are differentially methylated in your control and test samples. You have input your file as a methylation call files, and you have done all the magic in MethylKit, and now you know what all genes are differentially methylated. The final file looks somewhat like this, which tells me about the start site or end site. This is the same base. So the strand of the methylation plus strand and minus p values and q values for statistical analysis, the methylation difference, and the identity of your genes. Now that you know the identity of your genes, what you can do is you can validate your data using different approaches. One such approach is methylation detection by PCR. For that, what you'll do is you'll purify your DNA, the same DNA that you used for whole genome bisulfite sequencing. It is better to take the same samples for avoiding any discrepancies. So again, you will bisulfite convert your data, you will quantify your data and assess the quality of your data. After that, you can go for either bisulfite sequencing PCR or you can go for methylation specific PCR. In bisulfite sequencing PCR, you are just simply bisulfite converting your DNA and using PCR to amplify your gene of interest and going for gel extraction and cloning your insert into a vector, and finally knowing the sequence using Sanger sequencing. In methylation specific PCR, what you'll do is you design 2 specific set of primers, one for your methylated allele and second for your unmethylated allele. There are 2 set of primers, methylated forward and reverse primers, and unmethylated forward and reverse primers. So this is the main difference between bisulfite sequencing PCR and methylation specific PCR, that in bisulfite sequencing PCR, you only use one primer, while in methylation specific PCR, you use two set of primers. So if you have 10 samples for methylation specific PCR, you will use those 10 samples with methylation specific primers as well as unmethylated specific PCR primers. If your sample is methylated, you will see amplification only in the methylation using methylation specific PCR, but not using unmethylated primers, and vice versa. So based on your approach, you can go for bisulfite sequencing PCR or methylation specific PCR. But one thing that you have to keep in mind using methylation specific PCR is that you need proper controls, proper methylation controls that will tell you that your system is working fine. So we'll be doing a hands-on session on designing primers using MethPrime for both bisulfite sequencing and methylation specific PCR. So this is the general pipeline. The primer designing step is the main thing to keep in mind while going for bisulfite conversion and PCR analysis. And once you have designed your primers, you conduct PCR using hot start polymerase. This is very important in bisulfite converted DNA, that you need a hot start polymerase. So you will run your PCR, you will visualize your PCR products on agarose gel, then gel extracted, and then cut the gel out and clone it into a vector and get it sequenced. And once you have get it sequenced, you analyze your sequence data like you do for normal PCR, but for methylated data, you need a different software like Cuma. So we'll start with the hands-on session on MethPrimer and Cuma. Quickly, now that from methylated data, I know this locus is differentially methylated. So what I'll do is I'll open NCBI and MethPrimer. Right. So these are freely available, both Cuma and MethPrimer. Methyl kit as well. So I'll search for my gene here. So this is the MethPrimer site. We'll go for go to MethPrimer and let's see if it is loaded or not. So this is my locus, FLC locus. I'll go for gene. So remember, when you are doing DNA methylation analysis, you'll go for the gene sequence, not the transcript sequence, because our starting material is DNA, not RNA. So we'll quickly go for a faster file. So this is my faster sequence. We'll copy this and paste this here. So we are going for first bisulfite sequencing PCR. I will pick up, I will click at this one and then use CpG island prediction for primer designing. So I told you CpG islands are the regions which have a higher frequency of CpGs. So we are using the CpG island prediction in designing our primers. So we'll submit and see. So it is saying no primers found. Okay. For MSP. So we will come back and select another locus for bisulfite sequencing. Time being, we can design primers for methylation specific PCR. Okay. This is my locus that I have input. It is telling me that this is the name of your sequence and the length of the sequences 4431 base pairs. And the criteria for using for selecting was, it is saying that the size of your island or the sequence should be more than 100 and the GC% should be more than 50, and observed upon expected value of GC should be 0. More than 0.6. So whatever area that is clearing this cutoff will be known as a CPG islands. So it has told me that there is one CPG island in your locus and it has designed primers accordingly. So this is my methylation specific PCR, methylation specific primers, and these are unmethylated specific primers. Set of primers I told you, M primers and U primers. There are 5 such primer sets you can choose based on specificity, the TM and the GC%. So this is MSP. So we will do it for we will search another locus. Um, I've already done this work. So I'm searching for one more locus to show you how to design primers for bisulfite PCR. So we'll clear all this. We'll search for this locus on NCBI. This is my locus. Again, we'll go for FASTA format of my sequence. I'll copy this sequence and paste it. Now I am designing primers for bisulfite sequencing PCR, and we'll again use CPG island predictor for designing primers. So this is my locus, and it is telling me there is one CPG island of length 240 base pairs, and it is also telling me the position. And it has designed primers based on CPG island. So it is telling me there are 5 primers, primer set 1, 2, 3, 4, and 5. So these are the primers. You can pick these primers and go for synthesis, set up your PCR, get your PCR product cloned into the vector and get it sequenced. Once you have get your product sequenced, PCR product sequenced, so this is the same thing. These are the locus for which methylation specific PCR are designed, primers are designed. So what I was telling you is that once you have set up your PCR, got your PCR product, got it cloned into a vector and got it sequenced, what you do is you will analyze your sequencing data. So for analyzing your sequencing data, you need a software. The normal bioedit software or some other software are not supported for methylation and statistics on methylation data. So this is QMA that we use for methylation data analysis. This is the homepage of QMA. You your submit your sequences in GenPack format or your FASTA format or plain format. One is your reference genomic sequence, and second one is your bisulfite converted sequence or your sequences that you have got after sequencing. So for statistical analysis, you need to click on show option fields that will also perform statistical analysis on your data. So you'll submit your data. You can see your alignment files that have been aligned to your reference genome. You can download your alignment files. You can download all the pictures that will show you your methylation pattern on the sequences. So let's quickly go for human software. So this is the homepage of QMA, quantification tool for methylation analysis. You will load your reference genome or a reference sequence for which you have done your your bisulfite conversion. You can get it from NCBI. So I load my reference sequence. and I will upload my sequencing file. So I have loaded my reference sequence as well as the FASTA file of all the sequences that have been sequenced. So this is the list of 16 sequences that were sequenced after bisulfite conversion. And this is my reference sequence. So I have loaded it and submit. So the length, it has given me the summary of information where it says that the length of your target genomic sequence is 453 base pair and there are total 19 CPGs. And for an answer, it has used 10 sequences out of 16 and it has excluded 6 sequences. We'll see why it has excluded 6 sequences. So it is telling me the position of CPGs, 26th position, 42, 49, and so on. There are total 49 positions. So you can see your data graphically and download your graph. See, it is telling me that this position is methylated. Also, you can see it has excluded these sequences because of mismatch or differences in the alignment length, or it is clearly seen that the percent identity is very less. So it has excluded all these sequences. So you can see this figure as well. Sequence wise, these are my sequences 1, 2, 3, 4, and it is telling me the methylated CPGs. So now you have done the data analysis of only one set of sequence. So if you have your treatment and your control samples, what you'll do is you load both of them. So I will show you how to compare your treatment and control samples. So this is a zip file containing sequences of both your treatment and your control. So in one you will upload this and one your previous file. So this is one, let's say this is my treatment and this is my control and this is my reference sequences. So I have also clicked on the more options. Now it will do the statistical analysis of my data as well. Since we have used my used treatment and control, so it will show me the difference between the two, statistical difference between the two. So now you can see it is telling me information of group 1 sequence, group 1 and group 2. Same position but different group of sequences. Again, it has excluded some sequences based on percent identity or mismatches. You can see the graphical representation. So at looking at this, you can say that at position 42, the group 1 sequence is more methylated as compared to group 2. Similarly, here it is not methylated, but it is only methylated in one of the samples. So now you can compare your test and treatment samples, test and control samples. These are the alignments. You can download your alignment files. You can download your statistic data. That's pretty much it. So now I've shown you how to quantify your methylation data. So till now, what we have done is we have analyzed our data, whole genome bisulfite sequencing data using methyl kit by using a basic methylation call file as an input. And what we got is output file that told us that what is the location of the methylated site in which gene. So using that gene, you can validate your WGBS result using PCR by designing bisulfite sequencing primers or methylation specific primers. And further, you can analyze your data or quantify your data using QM. So this is all we have done. So thank you very much. I would like to acknowledge my professor Suman Paul for her immense support and ICAR, NASF, NL University, IOE funding. I would also like to thank CSIR for my JRF fellowship and Department of Botany for providing all the facilities in the department. And thank you, Zakir Hussain Delhi College Botany Department for inviting me to share my research on this platform. So thank you. Any questions?
Thank you, Ms. Pratima. It was a very detailed and illustrative hands-on session. Thank you for using this offline tool. R is a very powerful tool, and these days research is almost incomplete without using R. Thank you for giving your hands-on session on R and its use in your research.
Thank
So
you, sir.
I
I'll be
request.
happy to take any questions.
Yeah. So I think there is one hand which is raised. So I will allow the participant to speak. Uh, this is Miss Rani. Miss Rani, I have disabled the mic, enabled the mic, and allow the camera. If you have any query, you can ask. Miss Rani. Miss Rani, am I audible to you? You have raised your hand.
Dr.
I think
Zisha,
she's
I
not.
think, yes, yes.
Yeah, she is not available. Okay, so we are already running ahead of time. So now I will propose a vote of thank for you, Ms. Pratima. Thank you very much for giving this nice hands-on session on R for your research work that is on methylomics. And thank you very much for joining us and preparing video for the event. So thank you very much. Hope that for the future events also you will be available and we will have any offline event and you could
Yeah,
show us
definitely.
in front of all of us. So thank you
Thank,
once again.
thank you, sir.
So now I take this opportunity to invite another resource person for the hands-on session, Miss Shubhangi Kala. Miss Shubhangi Kala.
Good afternoon, everyone. Is my video visible?
You are audible, but your video is not.
Actually, I'm trying to switch it on.
Okay, no problem. So you can try, and now I shall take the opportunity to introduce you, and in between you can allow and you can switch on your camera. That would be nice to see you also. So Miss Shubhangi, yeah, now you are visible. Good to see you, Miss
Yeah.
Shubhangi. Shubhangi. So
Yes,
I
sir.
take this opportunity to introduce Miss Shubhangi Kala. She completed her undergraduation degree from Sri Guru Tegh Bahadur Khalsa College, University of Delhi in 2017. After her graduation, she pursued her master degree in botany honors from botany from Savitri Bai Phule Pune University, Maharashtra, and completed in 2019. During her post-graduation time, Shubhangi worked on screening and characterization of mutation in BADH2 allele in aromatic rice varieties as a part of her MSc dissertation. In 2020, she joined as a project fellow at Plant Molecular Genetics Lab, Department of Botany, University of Delhi, under the guidance of Professor Suman Kanpal. Ms. Shubhangi's area of research interest involves delineating the effector biology of phytoplasma affected selected crop taxa in India, for which she utilized MS-based proteomic methods to unravel the molecular players involved in plant pathogen interactions. Now I request Dr. Ruchi to play the hands-on session. Thank you.
Before playing the hands-on session, I just wanted to inform you all that this particular hands-on session is in 3 segments. So we will be playing 3 videos one by one. First video is comparatively shorter for say 12 minutes. And then we have 2 more videos in continuation. So I'll be sharing my screen. Please confirm, Dr. Zishan, if my screen is visible.
Yes, it is visible.
Okay.
It's gone.
Yeah, because I forgot to include the sound. So just a moment, please. Is it visible now? And
Good afternoon, everyone.
is it
I am
Yeah, it
audible
is
as
Shubhangi, and
visible
today
and
I will
sound is
be
also clear.
discussing about
okay? Thank you very much.
quantitative proteomics in plants. So before jumping to it, I would like to discuss why it is important or why it is an important area of research. So as we know that all the expressed protein molecules of a biological system is termed as its proteome. And like transcriptome, the proteomes of different cells in the same organisms can vary greatly and are capable of adapting to internal and external stimuli by modulating the protein expression levels. Although the protein synthesis is directly dependent on the expression of a corresponding mRNA, but there are factors that correspond to there are factors beyond transcription concentration that um, contribute greatly to protein expression levels and the landscape of the cell. In contrast to one gene, one protein, and one function paradigm, the cellular complexity arises from many mechanisms that expand the molecular diversity beyond the protein encoding genome that we have. These mechanisms can be like RNA editing or alternative splicing, and at transcription level and at translation level, there are alternative start or stop codons. Lastly, um, lastly, a high degree of diversity and diversification is introduced by post-translational modifications, that is like uh, pho- phosphorylation, acetylation, etc. So it is essentially the reason that living organisms, living systems are able to adapt to their changing and conditions. Specifically, plants achieve this by multiple mechanisms that modulate the quantitative composition and diversity at the proteome, at the molecular level. And this molecular diversification at the proteome level is highly pronounced, is particularly pronounced. Um, Therefore, which expands the repertoire of functional modules inside the cell. So, in the study of proteome began evolving in 1930s with traditional quantitative methods such as ELISA and Western blotting. Then in 2000, it gained momentum with the major advancements in 2D gel electrophoresis and MS. With the rise of mass spectrometry, there was a significant shift from qualitative to quantitative MS-based proteomics, and this has significantly contributed to unraveling many um, mechanisms like cellular signaling networks, and then protein-protein interactions, and even mechanisms behind plant pathogen interactions. Overall, as we know that MS-based proteomics has the ability to reveal the qualitative and quantitative state of a proteome, and can also provide insight into the biochemical state of the cell. What basically MS proteomics does is that they use the quantitative data for peptides obtained through LC-MS/MS and measure the relative protein abundances across the samples. We'll see this now in the next slide. So broadly, quantitative proteomics are of two types, top-down and bottom-up. Top-down is where the intact proteins are measured, and bottom-up uh, are is the proteomics approach where peptides are measured. Peptides are measured as surrogate for their protein of interest. And for today's session, we'll be focusing on bottom-up approach. In a typical bottom-up approach, the protein is extracted from the sample and it is trypsin digested into short peptides, which are then separated onto liquid chromatography. As the peptides elute from the chromatogram, they are subjected to electron ion spray and sorry, they are subjected to electron ion spray and they are directly sprayed onto the mass spectrometer, where there are two levels of MS takes place in tandem, MS1 and MS2. At the first level here, the mass analyzer measures the mass to charge ratio of peptide molecular ions, and at the second level, um, these peptide ions are fragmented into specific peptide ions, which this MS2 detects as the MZ values. So the specific fragment ion pattern of each peptide ion together with its MZ value enables a confident identification of peptides present in the sample. And then the identified peptide sequences can be mapped to proteins, and the signal intensities of either the peptides or their fragment ions can be used to estimate relative changes in the abundances across the sample. Going in detail with bottom-up approach, traditionally they are divided into two types, that is global and targeted proteomics. Global proteome profiling is the most commonly used approach, and to analyze a proteome, it is generally preferred for comparative analysis of two or more proteomes. Broadly, global proteome is also divided into gel-based and gel-free approaches. In gel-based approach, it includes 1D, 2D, and 2D DIG electrophoresis. Then coming to gel-free or shotgun proteomics, this has the ability to identify thousands of proteins per run. And gel-free or this approaches include many types of um, methods like SILAC, ICAT, iTrack, and much more. In these, again, in gel-free proteomics approach, the protein mixture is subjected to trypsin digestion, and digested proteins are chromatographically separated and analyzed by MS. Then moving on to targeted proteomics. Um, Targeted or selective proteome analysis is the next major class of proteomes that was grouped on the basis of coverage of proteins. This targeted proteomics is mainly used for two reasons. It is mainly used for two reasons. For identification of interacting proteins or the proteins of interest, and then identifications of PTMs. Generally, the proteome profile focuses on identification of selective proteome or the selective proteome or group of proteome through motif or through PTM specific stains or targeted MS assays. And then again, it is divided into gel-based, affinity and reactive chemistry-based and MS-based. Broadly on gel-based, they are, for gel-based, the analysis is carried out using protein-specific or PTM-specific staining procedures that can be visualized after staining with PTM-specific stains, and which this staining procedures are usually followed after a global profiling using 2D electrophoresis, 2D gel electrophoresis. And then the MS-based or targeted approach utilizes the MS acquired data for peptides associated for these proteins in either targeted manner or in that it includes SRM, PRM, or MRM, or in a it can be either in a targeted manner, or it can be comprehensive data independent acquisition, which is DIA and SWAT. In comparison, if you say global proteomics, in comparison to global proteomics, the targeted proteomics provides a consistent and accurate quantification even at low abundances and in complex mixtures. Although targeted proteomics is mainly limited to a few dozen predefined proteins to run per run, it is still very sensitive and quantitatively capable, making it making it well suitable for a hypothesis-driven research, if you say. So now, with this basic knowledge, I think we can now get on to a more of hands-on session, where I will tell you how to utilize data obtained from label-free quantitative proteomics. Again, it is a global proteome data where we have tried to understand the mechanisms involving in plants that have undergone an infection by phytoplasma. So the plants here are sesame, and the infection is by phytoplasma, one of the phytoplasma strains, and we'll be dealing how to analyze the data based on their raw files.
Sir, I think you can now play the second video.
Yes, we are working. Dr. Ruchi? Just a moment.
I am playing in a moment, please.
Dr. Zishan, is it visible and audible?
It's not audible, it's visible.
Okay, okay, okay.
And Perseus softwares.
Is it all right now?
So
Yeah,
now that we have learned the basics of
yeah,
quantitative
all.
All
proteomics,
right.
we will see how to analyze the data and analyze the data obtained from LC-MS. So for today's session, we'll be dealing with label-free quantification data, and that is LFQ, and see how to analyze the raw files using MaxQuant and Perseus softwares. Before beginning, we need to download both of these softwares from the online website. So this is how you can get the software: Google MaxQuant, and fill this very basic form and download it from here. After your software is downloaded, you can install it and open the application. And this is how it should look like. So MaxQuant is a quantitative proteomics software package. As you can see, it is designed for analyzing large-scale mass spectrometric datasets, which enables proteome-wide protein identification and quantification. Several labeling techniques as well as label-free quantifications are supported with this software. And luckily, MaxQuant is freely available and can be downloaded just like how I showed you. So as you can see, there are certain tabs given here. We'll go through each one by one. The first thing is we have to load our data. You can either load it individually or you can load the entire folder from here. So we are going to load our data individually. We have 6 files, 3 replicates for healthy and 3 replicates for infected. And this is how it looks like after getting uploaded. Then the first thing is the assigning the group parameter group. If you want to assign different parameters to your data sets, then you can assign a different group number to each of the data set, or you can leave them all under the same group like here. If suppose I want to assign different parameters to all the infected files, I1, I2, and I3, then I can set a parameter group that is group 1, like here. And then I would later on, when I would go to groups assigning parameters to each of the group, I can assign them differently as per my experiment. But for today's data, we are not going to do that. We'll keep all these parameters same for each file. So that's why it shows group 0. Then the next important thing is to assign set the experiment. So basically, everything having the same name in this experiment column will be combined in the output. So technically, all the replicates should belong to different experiments, because otherwise all their intensities will be combined. So I'll show you how to set the experiment. For example, we will set it as control 1, 2, and 3. And this we'll set it for our convenience. That's how we are naming it, so that we know which file is which. Okay, sorry. This should be different. So moving on to other columns, like here PTM is kept false because our data does not have any PTM enriched experiments done on it. For example, it's not phosphorylation enriched data or acetylation enriched data. So that's why we are going to keep it as it is. Then we can move to group specific parameters. Here type is used for labeled proteomics parameters. But since our data is label free, so we are going to keep it as default. Standard is the default one. You can put it to reporter INMS2 for if I had a TMT label data, or a reporter INMS3, mass spectrometry 3, if I have other my other things. So for if I keep it standard here and put multiplicity 1 means no label. If suppose I have SciLab data, then I can put 2 and 3 multiplicity as per my experiment type. And then I can assign which light and heavy labels are in my data. So since this is label free, we are not going to do anything in this. We'll keep it as it is. Then we'll move to digestion. Here our data was digested by trypsin enzyme before getting onto LCMS-MS. So as this is the default here as well, trypsin digested. Otherwise, in other experiments, you can label it as per your data. Then we'll move to modifications. Here, these are the default parameters, default modifications set here. But if we want, we can also add few modifications that we would like to detect in our proteome. For example, if I want to detect acetylation at lysine residues, I can add it here, add it in the right-hand side column. And if I suppose want to detect ubiquitination at lysine residues, if I want to detect phosphorylation at serine, threonine, and tyrosine residues, so I can. Then there is this is for sumylation. So I can all add any of these parameters that I want to detect in my data. And it is advisable to add one modification at a time. Otherwise, the analysis will take, I don't know how many days. So for now, we are just going to remove any of these modifications. But you can add one at a time and then run your analysis to detect any of the things you want. Then comes to label-free quantification. This is the inbuilt parameter here. And since it is our data, so we are going to put it as it is. And in these, you can tweak the parameters as per your experiment again. We can also keep it as it is. But if you want to tweak it, like we can add, do LFQ minimum count ratio as 1 if we want. So this means it means that one peptide ratio is sufficient to report the protein ion. That's preferred, but that is okay if you want to do it, but you can also just go with the default parameter here. Then there is instrument tab, and it's usually advised not to change anything in this, as it is, there's no change that you need to do. Then we can directly go to global parameters. It also had some subcategories here. So the first is sequences. Here, this is where you add your FASTA files of the reference proteome. For example, my proteome is for sesame plant, and I will add the FASTA files downloaded from Uniprot of the sesame proteome, and I will add it. I can also add multiple proteomes as a reference if I want to. For example, if I'm adding a sesame FASTA file, and then along with it, I'm adding the phytoplasma that's the infection. So I can also add this FASTA file along with this. And this is how it looks like. Then there are other certain things that you can see here. You can also, you can also change these, but it's usually preferred not to. For example, minimum peptide length. It should not be too short, otherwise the peptides will match to many different proteins. Then is identification. Here, by default, peptide and protein FDR, that is false discovery rate, is set as 1%. You can change it, however, but this is the default parameter. Then again, this is protein quantification, is label minimum count ratio. Here again, we don't have to do anything. This is for the labeled quantification. And then, okay, these are the list of tables that you would want in your output file. This is sufficient for now. We can keep it as it is. And then finally, if you want to change where your folder locations should be saved, for example, if I want to save the output files in a different location than where my raw files are, then I have to specify where I want to put it. Otherwise, if you do not put any location, it will directly save all the output to where the raw files are. The rest, all the parameters can be left as it is. And then performance is where you will see all your analysis steps happening. Okay, this is stuck at visualization. Okay, yeah. So here, once the analysis starts, you can see every step that is happening at the moment. And then visualization is where you see your FASTA files, your proteome raw files. And these here you can add any modification you want. For example, it did not have nitrosylation earlier, so I added it on my own from this configuration option. And that's it. So coming back to the first tab, here, this is the number of processors, and you can also see what it says here. It's like you have to have minimum, at least minimum of 2 GB RAM in your laptop to run this software. But again, if you just have 2 GB, it is going to take a lot of time. So it's better to have more than that. So I can, if suppose this laptop has 12 GBs of RAM. So I have the maximum I can go here is number of 6 processors, but I'm still going to stay at 5, and then I can start my analysis here. So this analysis should take at least 4 to 5 hours at least, and then I'm not going to run it right now. We are then directly going to the Perseus software to analyze the output files of this. So again, for that, you have to go to the same website. From here, you can download it like the same way you downloaded Max Font. And that's it. So after you have installed it, this is how it should look like. Wait, I'll close this. Right. So this is how it should look like. It is another software platform that helps in interpreting protein quantification, interaction, and post-translational modification data that we just saw, how we can detect that. And as you can see, Perseus is another user-friendly interactive workflow that provides us comprehensive statistical tools for omics data analysis. So once our raw files are read from MaxPoint, we can use this to filter our data and put statistical tests and get a better data visualization via this software. Now, the first thing is, this is the loading tab, then this is the processing tab, and this is the analysis tab. So first we have to do is load our matrix. We will go to where our MaxFont analysis was saved. I'll show you what it will look like. So these are the folders that you will get at the end of your analysis: H1, H2, H3, I1, I2, I3, and combined. Our important is combined, where we'll go to text and go to protein groups. This and this, as you can see, contains 5 feature tags, that is main, numerical, categorical, text, and multi-numerical. And this is where you can choose the information you want to have in your matrix as per your analysis. And in the main tab, the LFQ intensity values are selected. These are the values that express the protein abundance in our data. For example, this is the values we want. And these values will be used for further statistical analysis. And here in categorical annotation tab, there are three different default parameters, as you can see. They will be further filtered out from the data, as you will see now. And you can add anything you want from this to any of these 5 tabs you want. For example, I can now add faster headers if I want to the text column, or any other thing you want, sequence length or peptide IDs here. You can add anything you want. I'll just keep this as it is for now. And this, all the identification data containing all the data you just selected will be shown in this data matrix. And as you can see, there's this one step you just performed will show here as a workflow. So any further downstream analysis you'll keep on doing, you can track all of the actions that are taken during this data processing and analysis. And this workflow can be saved later on as well. So if you see here, all the columns that we had selected are now shown here. Pasta headers I added in the last. So this is also here. So these are my list of proteins that are detected. As you can see, 1408 number of proteins. Now the first thing that we have to do is data filtering. For that, first we have to remove the unnecessary or incorrect protein identification from the main data frame. So what we will do is filter rows based on categorical column. And so it's in categorical column. I told you the 3 default parameters that we'll get rid of as a filtering process. So only identified by site, all the values having a plus sign are selected here, and we will remove these rows and the matrix will get reduced. In another case, you can also keep the matching rows, but in this scenario, we want to filter it out. So we are choosing this option and OK. So as you can see, 8 of the items got removed. Now another same thing we will do with reverse. All the column values with plus will get removed from this column. And again, we're going to keep it as it is. OK. As you can see, more proteins are filtered. And then last, potential contaminants. If you want, you can also keep the contaminants. It completely depends on your type of experiment, but here we are going to remove it. And then this is it. The final is by 1380. And now what we can do is the next step is now we will transform our data. So basically the problem is that the range of expression values can vary more than 10-fold. So the expression values can be log transformed in order to facilitate the calculation of the protein expression fold change. So I have now, as we can see now we can, so this is the selected, we can change the log value here, but log 2 is the default and it's okay. And the columns that will be log transformed are selected already, which is the intensity values of control and infected. And so as you can see, the values have been transformed. Now in order to group the conditions, now we know that there are 2 conditions of control, 1, 2, and 3, but for the software, it's 6 different conditions actually. So what we have to do is group the conditions that are to be compared in this quantitative analysis. And for that, we will go here. And in the row name, that is group 1, we will just put these values put them as under the same name for all the values that are under the same group. So remember in the initial max quant, we had given them all different names, but here we have to place them in the same group. Like so. So in this way, these two conditions can be compared against each other. And this is how the statistical test will take place on these two grouping categories. Now, next, one of the most important filtration step is filtering based on the valid values. And as we can see, there are a lot of a log transformation that has converted from 0 to NaN values. This actually non-assigned number, NaN values, only corresponds to the expression values which originally equal to 0. So when proteins were not detected by the MS, in order to define the level of stringency in your analysis, we need to at least define the minimum number of valid values accepted for our analysis. And this totally depends on our kind of experiment. We have to think what kind of filter we need to put for this. For example, it says minimum valids in number for minimum 3 values that are in total. Minimum 3 valid values that are in our 2 groups, or at least 3 minimum in each group, or 3 in at least one group. So this is where you have to actually think what suits best for your data. You can also put it in terms of percentage. 70% of valid values, again, in at least one group, that is in healthy or infected, like control or infected, or in each group, or in total. So for current thing, we can do minimum 2 values in at least one group. Okay. So as you can see, major chunk of filtration took place at this step. This is the deciding filter that can increase the confidence of our data. Okay, now that we have transformed the LFQ intensity values to log values, and then we have also filtered our values on the basis of valid values. Now we can impute the missing values using normal distribution. Here we can go from imputation, replace missing values from normal distribution, because we need to run down the other analyses, and we can now, we have to remove these NaNs for the further analysis. And this is again the default parameter set up for replacing values from normal distribution. And that is done. And you can see how all the steps are recorded as a separate matrix. You can go back to any matrix or any step you want. And you can also check what happened in before, what happened in this step, or what happened, or what parameter you set up in this step. You can always go back and trace your analysis. And okay, so here we are. You can also name the matrix you want. If you want to rename it as per your wish, for example, if I say, I can do that. I can change it as per my way. So, okay, now that we have transformed everything and filtered everything, you can assess the degree of correlation between different samples. For example, now if you come to the analysis group, we can and try to understand to what extent the replicates are similar or dissimilar to. So
Shubhangi,
Yes, sir.
can we
So
play the
actually, I
third
do.
video?
No, sir. Actually, this video is not complete yet. I think.
Completed here. Okay, okay. Let me check.
Yes, sir. It got cut in
I'm
between.
sharing it again. I'm sharing it again. There is some, I think, problem from my end. I'm sharing it again.
Okay, ma'am. No problem. Is generated.
Can you see Shubhangi and
You
Dr.
can also alter this
Zishan?
if you want.
Yeah, it's visible.
Yes,
and
ma'am. It's
visible.
transformed the LFT
Is it
intensity
audible?
values
Yes,
to
ma'am.
log values, and
Is
then we
it,
have also
means,
filtered our values on the
should
basis
I
of
play it from little means
validity, valid
गया था
values.
या कितना रह गया?
Now
Because
we
I
can
was
impute the
I
missing
was not
values
in track.
using normal
Video.
distribution. Here we can go from
Where was it?
imputation,
Ma'am, maybe you
replace missing
can.
values from normal
Ma'am, maybe you can
distribution. because we need
fast
to
forward to 2 to 3 minutes.
run down the other
Okay.
and you can. Okay, so more again, one thing that you can do is do a hierarchical.
Is it fine now?
No, you can do a
Shubhangi?
histogram
No,
here if
ma'am.
you wish to.
Just, ma'am, just 1
Here
minute. No, no,
is
no, ma'am.
go to test.
You've gone too far.
Okay.
Ma'am, maybe you can stop at 25 minutes. It's, I think, okay with 25. Okay, let 24
A
maybe
little
now.
before, I guess.
Even
Yeah,
25 seems too far.
Is it?
I think a little before.
You
Yes,
can
ma'am.
color it
Is it fine
differently
now?
if you wish to.
A little before.
A
For
little.
example,
Yes.
it will
20.
help us to
I think
calculate
that
and
was the
identify
place.
these possible.
This was
We
the
can go
place.
to multi-scatter option.
So I
Okay,
have started
now
with 22
that
minutes,
Yes.
right?
we have
Okay.
transformed
Yes,
everything and
ma'am. This
filtered
is fine.
everything, you can assess the degree of correlation between different samples. For example, now if you come to the analysis group, we can and try to understand to what extent the replicates are similar or dissimilar to each other. We can go to multi-scatter option, and it will help us to eliminate the data sets that has outliers, and it can help us to calculate and identify these possible outliers. So now we can just click open OK here. And as you can see, a multi-scatter plot is generated. And you can also alter this if you want. And you can color it differently if you wish to. For example, like this. If we do it, okay, a better color maybe. So right, we have done it this way. And also we can put the Pearson correlation values, that's the most commonly used. And this indicates the calculated correlation between the data sets on X and Y. As you can see here, maybe it's not visible in this, but here you can see, okay, wait. Okay, this is fine. So here, this is healthy, control 1, 2, 3, control and infected, and then likewise control and infected. And as you can see this, the closer is your correlation value to 1, the higher is the correlation between two data sets compared. Next, we can also go to statistical analysis, that is for determining the variation between proteome of 2 or more conditions. Okay, so again, one thing that you can do is do a hierarchical, no, you can do a histogram here if you wish to. It will also generate a histogram where you can see where most of your data lie among this range. The intensity, LFQ intensity of your data is lying in majority of which range. Okay, now coming back to the statistical analysis, we can go to this. Yeah. So for this, we need at least 2 or more conditions, and it is necessary not only to just identify the proteins in a sample, but it's also to perform the statistical test to determine if the changes that we are observing are experimentally in the proteome, are they also statistically significant? So what we can do here is go to tests and sample to test. In this analysis, we are going to use 2 sample test, and here we have selected student t-test, and in this we can do Benjamin Hirschberg FDR, and we can also change to 0.01 or 0.01 or 0.5 is fine for now. And as you can see, there's another 2 columns that are now generated. So these are all the proteins that are significantly, they are statistically significant. No, now, so these are the 100 proteins that are significant in this data. So what we can do is now again, we can filter rows based on categorical column and select only the proteins that are significant. And we can now keep the matching rows that are having a positive sign, and we'll reduce the matrix. So now this new matrix that we have has only 100 proteins having all the significant proteins. And now we can also make a heat map out of these. What we will have to do is, first we have to normalize LFQ data to Z-score. And that's it, just press OK. And then we can just directly click this option, that is hierarchical clustering. That will cluster data using Euclidean distance method. Here, yes, Euclidean distance method, and that's it. Okay. As you can see in other tab, the heat map is created. You can again alter this, modify the row clustering width, name width, or header width. You can change it as per your preference. And you can define row clusters. These are the 2 different clusters that are here. This, the upper cluster is having proteins that are down-regulated in infected and upregulated in control, and then vice versa. The second cluster is the proteins that are upregulated in infected and down-regulated in control. And then we can also modify it from here, change the color that is there. And then here you can see the clusters, 2 clusters that are different in their Z-score values. This is the cluster 1, and this is the second cluster. And now you can also export this heat map to whichever folder you want. It can be in any of the formats. And you can also export all the other, let's say, multi-scatter plot or histograms via this option if you want. And in the end, it is always advisable to keep saving this session so that save this entire session so that whenever you want, you can go back to see the steps you did for the analysis. And you can also save a specific matrix if you wish to. For example, if I want to save the list of all significant proteins, so this one is the matrix for that, I can specifically go and export this matrix. It will be exported as a text file. I can save it in any of the folders, and it will be a tab separated file, which I can later use and modify and analyze in my Excel sheets.
Yes.
Uh, yes, Dr. Zishan, I'll be playing the third video.
Yes,
Just
ma'am.
wait for a moment, please.
And so this is one
Dr.
of
Zishan,
the
is it visible and
standard and
audible?
basic ways to process and
Yeah, it's fine.
analyze
Yes, ma'am, it's
your
visible and
raw
audible.
data obtained from MaxFont. And if you remember that I had told you, you can also add any kind of modifications that you want to detect in your proteome at the MaxQuant level. So if you have selected one of the modifications, it will get generated into the output file. And we can also use that to analyze the results obtained. All you have to do is now, if I will start, um, all you have to do is instead of protein groups text file, you have to upload the text file for specific to that PTM. For example, if I will show you here, so suppose this is my phosphorylation data. I'm going to combined folder, text folder, and now a specific file for phosphorylated sites will be generated here, and I have to just insert this. And now LFQ intensity won't be shown here. You will have to upload intensity, just intensity. Okay, intensity control 123 when infected 123. You will select this in your main option and FASTA headers in your text if you want. And okay, that's it. So as you can see, another matrix has been generated, same as the one that was generated for LFQ intensity. And now you can run the same analysis, exactly the same steps that you did here. And you can do this for your PTM data and shortlist the list of proteins that were modified. And that's it for Perseus. And now that after you have saved all your data and downloaded it in text file, all you have to do is to calculate the fold change, you have to subtract the differential. You have to subtract the um, intensity values from one another, and you will get the fold change values, and then you can decide the proteins undergoing differential abundance. And now what we'll do is try to analyze the list of proteins that are differentially abundant by using some of the online software tools. One of them which we'll be using is David. Okay, so you just have to Google David and David software, and this is the site that will come up. This is database for annotation, visualization, and integrated discovery. This is a bioinformatics resource that can help you to extract biological information and meaning associated with the large list of proteins or genes that are differentially regulated in our quantitative analysis. However, there are many other softwares available as well, but at the end, they all adopt a common core strategy is that they systematically map a large number of genes and proteins in a list to the associated biological annotation, for example, GO terms, gene ontology terms. And they then statistically highlight the most overrepresented or enriched biological annotations out of the entire list. Because enrichment analysis is a promising strategy, it increases the likelihood to identify biological processes that are pertinent to the biological phenomena under study. For example, here we are trying to study the plant pathogen interaction. So it will be able to detect all the proteins that are enriched in this interaction. So for this, for this software, how we'll go about it is we'll click on Start Analysis. And in the upload column, we have to either paste or directly upload a list of proteins or genes that are of interest to us. For example, any list of differential genes from our analysis. Then we have to select the format for the list that we are providing. It can be either a UniProt ID, it can be a gene ID, interest gene ID, it can be GenBank and session, or wait, it can be also a UniProt ID. Then the third part is you have to select the type of list. Either you are uploading a gene or protein list, or you're uploading a background list. So David usually can automatically detect the background list even if you don't manually enter it. Background list is the database of list where it will be detecting the enrichment from. And then you have to submit it directly. But for here, to explain this software, I'm going to just use the demo list provided here because it makes it easier, and I'll be able to explain all the functions of this software better. So this demo list is of Homo sapiens having 149 entries. And from here, we will select functional annotation tool. And as you can see, the result contains several different categories, and you can also expand each category you want. So for example, we are going to look at gene ontology, and it has several different searches. So it has BP, CC, and MF. BP is a biological process, which will tell us what biological process the annotated protein performs. Then there is CC, that is cellular compartment. That means where the gene or the protein belongs in the cell. And MF is what function it performs inside the cell. And then, as you can see, it has numbering 1 to 5, 1 being the highly broad term annotated to the gene. It's very broad. And 5 being very, very specific term. So as I'll show it to you here. So these are the list of annotated terms of the enriched genes or list of genes we entered. So as you can see, the term that they are provided here is signaling, growth. They're very basic, very broad. While on the other hand, if you go to number 5, it becomes very specific. Yes. So for example, it has neurogenesis genes or innate immune response in mucosa, defense response to fungus, and then antimicrobial humoral response, antibacterial humoral response. So it is very specific in comparison to the one we saw in the first term. Then there is geoterm BP all, which means that it will contain both very broad and specific terms entirely, as you can see here. And then there is BP direct, which has BP direct has all the terms that are directly annotated from the database, and it is also the default term that is selected by David. So if we see this BP direct in particularly in detail, we can see, right. So we can see that it has many columns, first being the name of the term, of course, and then RT, which stands for related term, meaning the terms, other terms related to the gene of our list. Then comes the gene number, meaning the number of genes that are involved or categorized under this specific term. And then you can also click on it, and it will tell you details about this in detail. It will give you a detail of every gene. Then also you can look at the p-value and Benjamin values. These are actually, they help you examine the significance of gene term enrichment with the Fisher's test. The smaller the p-value, the more significant they are. And however, the default is 0.1, but you can, a user or we can easily set different levels of cutoff through this options here. You can put a different value here. If suppose I want to put 0.01, and if I want to include FDR, then I can rerun here. It will give me the result with my specific options. Then, as you can see, these terms are also arranged in the decreasing order of significance. Down the list, you will find terms that are less significantly enriched. Then, apart from this, if you come back to this tab, you will see that there are many other things to discuss. But if you go to pathways here, again, all the default terms are highlighted in red. So the KEG is one of the default categories selected. So if we select on the chart, it will directly tell us all the proteins or genes that are involved in different pathways. For example, if I select MAPK signaling pathway here, so this is the entire signaling pathway, and all the red stars tell us the genes or proteins that are present in our differential abundant list, and they are annotated here. I can do this. So again, this is the list of all the proteins in the pathway, and red ones are the proteins that are present in our list of differential genes. I can also download this photo, but this photo will not have, the downloaded version will not have the stars on this. You will have to manually add it later on or annotate it with any other editing tool. Then now coming to functional annotation clustering. This is essentially a tool that condenses the input gene list into smaller and organized clusters. So for example, here we have 38 clusters, each having an enrichment score and list of proteins that are, and list of terms that are categorized under each cluster. So enrichment score is essentially trying to assign significance to how significantly, how much enriched it is in your data. For example, it has 4.5, is just 3.2. So as you go down, the level of enrichment is decreased, and a cutoff is usually around 1.3. Higher than that is something you will call interesting, or you will call highly enriched in your data. So for example, this second group of cluster, annotation cluster 2, contains mostly defense related proteins. This cluster usually is particularly containing binding proteins. So what you can do is here also go to this chart. You can select this and it will tell you for example, the cluster had 7 terms. So these 7 terms are in association with how many of the terms present here. For example, it will give you that all the 7 terms are associated with ribonuclease A family member, and they are also associated with WNT family members. So this is how you can also see your data being represented, and you can of course make sense out of it. And that is it. Again, there's protein domains. You can check all the domains that are present in your differential proteins. And that is it for now. You can also again use other softwares like MapMaker, Panther, or any other data software that supports your data. You can go ahead with it and carry out the analysis. So it's done for now. In my opinion, we are.
Thank you, Shubhangi.
Thank you so much, sir. It's a pleasure to be presenting in front of all of you.
It's a pleasure for us also to have you in our event, and thank you for preparing this long video and giving a detailed analysis. So the session is open for the question and answer session. Question and answer. I think there's one person who has raised the hand, Dr. Abhishek Rai. So I'm allowing the mic and camera for you. So Dr. Abhishek, you can ask your question.
Thank you, sir. Sir, I wanted to ask ma'am, thank you, ma'am, for your informative presentation. Can we use this Perseus software for subtractive proteomic screening, like to remove the redundant proteins, etc.? Like sometimes you get duplicate proteins as information. So to remove them, subtractive proteomic screening?
So actually, personally, I have not dealt with such a data, but in my knowledge, I think you can use this Perseus software for any other omics data analysis as well.
Okay.
So,
thank you.
Yes, sir.
Thank you, sir. Thank you.
Thank you. Thank you for your question.
Thank you.
Is there any other question? All right. So I propose a vote of thanks for Ms. Shubhangi Kala. Thank you for joining with us, and thank you for sparing your time with us. So we hope that we will like to have you in our offline events in future also. So thank you for your support. So now I request Dr. Ruchi to proceed further. Thank you.
Thank you so much, sir.
Thank you, Dr. Zishan. Now it is time for last lecture of today's session. We welcome our international speaker, Dr. Ravi Gupta. Dr. Ravi Gupta, can you hear me?
Yes, I can hear you.
Ravi, kindly switch on your camera, please.
Yeah. Okay, so I have already turned on my camera. Can you?
But I can't see you.
Just a moment. How about now?
Not yet.
Not yet. Okay, just a moment. So am I
Or
visible now?
Dr. Zishan, is it
I think
Ravi's?
we can proceed further because we are
Yes,
already
yes,
running
yes,
ahead of
yes.
time.
Okay. So,
Good afternoon, sir.
good afternoon, Ravi. And it is our privilege to introduce you to our speakers. And I welcome you. And you are alumnus of our department only, Department of Botany, University of Delhi. And I would like to introduce Dr. Ravi Gupta to our participants now. Dr. Ravi Gupta, he's an assistant professor in Cookman University, Seoul. He obtained his PhD in plant physiology and biochemistry from University of Delhi, India, and he has spent more than 5 years working as postdoctoral fellow and research professor at Busan National University, Busan, Korea. Dr. Gupta is the author of more than 90 scientific publications with a cumulative impact factor of more than 250. And also there are two books, edited books to his credit. He is the recipient of many prestigious national and international awards, including Ramalingam Swamy Re-Entry Fellowship, Ramanujam Fellowship, and ITS Fellowship from Government of India. And also he is recipient of Korea Research Fellowship from Government of South Korea. Dr. Ravi has also been a member of New Phytologist Next Generation Scientist meeting held at Norwich, UK, and the 10th Max Planck Summer School organized by Max Planck Institute of Biochemistry, Germany. He has gained almost 12 years of experience in the field of proteomics and is currently utilizing advanced proteomics tools for deciphering the biotic and abiotic stress signaling in plants. Dr. Ravi, I welcome you to our workshop and conference. Thank you very much to be a part, and over to you now.
Thank you, ma'am. Thank you for the nice introduction and for your kind words. And it's a great pleasure for me to be here. And I must, of course, thank the organizers, especially Dr. Ruchi, for giving me this opportunity and for conducting this event. Okay, so are my slides visible to you?
No, sir, the slides are not visible.
Okay, just a moment. How about now?
No, no, Dr. Ravi, it is not, your slides are not visible. Are you trying to upload or you are sharing your screen?
I'm sharing my screen.
No, but it is not visible.
I suggest you can rejoin.
Okay, let me rejoin. Okay, I'll try sharing my screens now. So how about now?
Yes,
Yes,
yes.
it is
Now
visible.
it is visible.
Okay.
You can give, you can share it in full screen mode, sir.
Okay. So is it okay now?
Yes, sir.
Okay.
Yes.
So thank you once again, Ruchi ma'am and Jishan, for giving me this opportunity to talk something interesting and to talk something that I'm following and I'm working from the last 12 years. Okay, so today I'm going to present on integration of labeled and label-free quantitative proteomic approaches to dig deep into the plant proteomes. And with the help of this presentation, I would try to introduce you the basics of proteomics, its history, and recent advancements. And since this is a workshop, so I'll not go into the details of my experiments, and I would like to provide you an overview of what is being done in proteomics these days and how proteomics has evolved in the last 3 decades. Okay, so starting from the basics, as Professor Deswal has already explained about the central dogma of molecular biology, I would also like to start with this backbone of the molecular biology. So, I think every one of us is aware of central dogma, as per which the DNA that constitute our genome is transcribed into mRNA, which is translated into proteins. These proteins catalyze various functions inside the cells to produce a set of metabolites that determines the function of an organism. The information contained in the genome of an organism is static and it can only predict the possible functions of an organism. On the other hand, the transcriptome is dynamic and it changes from time to time depending upon the external and internal factors. However, even this transcriptome can only predict what can happen because of the lesser correlation between RNA and proteins. So proteins are the functional players that catalyze the metabolic reactions, and thus changes in proteome reflects the changes in metabolome or phenome in a better way. Okay, so let's understand this with an example of caterpillar and butterfly. These two are the same organism, yet their appearance, their functions, and their behavior are quite different. And sometimes people can even think that these are two different organisms. I myself, as a child, I was also thinking that these are two different organisms. I was quite scared of the caterpillars. I used to love the butterflies. So although they are having same set of genes and have identical genome, the differences in their life forms are because of the changes in their proteins and the proteins that they express. So you can just imagine what the changes in proteome can bring about. So what actually proteomics is? So proteomics is defined as the protein complement of a cell or tissue at a given condition at a particular time. And the term proteome was first coined by Mark Willikins in 1994 as an analog of genome. So Mark Willikins, he was actually a PhD student in 1994, and he was writing his thesis, and he was kind of highly irritated in writing protein complement of a cell in his thesis every time. So as an analog of genome, he coined the term proteome, which is now well accepted. So proteomics basically includes the accurate identification, quantification, analysis of protein modifications such as post-translational modifications, protein-protein interactions, and interaction of proteins with other biomolecules such as carbohydrates and fats, and analysis of biological activities of proteins. And one of the major questions that we try to answer through high throughput proteomics is how these proteins change during a biological response. So proteomics experiments can be broadly classified into discovery proteomics and targeted proteomics. Discovery proteomics is driven by exploratory investigations to develop a protein catalog present in a sample at a particular time, while targeted proteomics deals with the hypothesis-driven investigations and look for the presence or absence of specific proteins in a sample. So majority of the proteomics studies performed in the last 2 decades are based on the discovery proteomics, and those studies aimed to investigate the type of proteins present in different biological samples such as leaves, tissues in response to any stress condition to develop the protein snapshot, what we called as proteome maps. So if we look at the typical proteomics workflow, it includes the lysis of biological samples with the help of liquid nitrogen, followed by isolation of total proteins with the help of mass spectrometry compatible buffer system. And the obtained proteins are then subjected to enzymatic digestion, such as trypsin, to convert proteins into a complex mixture of peptides, which are then separated by liquid chromatography or LC and identified by mass spectrometry or MS. So MS analysis generate a set of spectra for each peptide, and these spectra are usually annotated to proteins with the help of different softwares and databases such as MaxQuant that Shubhangi has explained in the last hands-on training session. So in the upcoming slides, I will try to introduce all of these steps briefly and how these steps can be modified to improve proteome coverage of biological samples, especially plants. Okay, so let's begin with the first and I think the most crucial step of proteomics workflow, the protein extraction. So the methods for protein extraction from different plant tissues are well established, and it starts with the grinding of tissue using liquid nitrogen to make it a powder, addition of protein extraction buffer which usually contain a buffer system like trays or heaps, and some detergents to solubilize the cellular membranes, and the subsequent steps include the centrifugation of the extract to remove the debris and to clarify the supernatant, and the precipitation of proteins from the extract using one or more organic solvents such as phenol or TC-acetone or methanol chloroform. So as per my experience, phenol method among these shows best results in terms of protein clarity and number of protein identification. So I usually either go with the phenol method or methanol chloroform method, but the requirement actually depends on the sample quality. So if there are lots of secondary metabolites associated with your protein samples, then you may have to go with the methanol chloroform precipitation method. If on the plant sample your is quite clear, I mean, if um, suppose it's an Arabidopsis, so just phenol method will work fine. So in addition, you may also go with a combination of these methods uh, to increase the proteome coverage, because there could be some proteins which can be extracted by phenol method, some by TC-acetone, some by methanol method. So if you will use a combination of all these methods, you will have increased number of identification and deeper proteome coverage for the plant samples. So once you have a protein extract ready, next step is to analyze these proteins. So in terms of proteome analysis, we have developed majorly two kinds of approaches, the gel-based and gel-free proteomics approaches. So in the case of gel-based proteomics, isolated proteins are first resolved on the polyacrylamide gels. So the term is gel-based, while in the case of gel-free proteomics, resolution of proteins on polyacrylamide gel is omitted, and the isolated proteins are directly subjected to trypsin digestion, which are then identified by the mass spectrometers. So in the case of gel-based proteomics, introduction of two-dimensional gel electrophoresis was a kind of major breakthrough, which allowed the resolution and simultaneous visualization of hundreds of proteins in a go. And it was developed in 1975 by three researchers independently, including Close, O'Farrell, and Shield. So two-dimensional gel electrophoresis or 2D includes the separation of proteins on two dimensions, as the name suggests, in which the first dimensional separation is based on the intrinsic charge of the proteins and is achieved on the IPG strips or immobilized pH gradient strips, while the second dimensional separation is based on the molecular weight of proteins and is performed on the SDS page. So after the completion of 2D, proteins can be seen as distinct spots on the gels, which are visualized by different staining methods such as CBB and silver staining. And identification of proteins that changes in response to a treatment or stress condition is done by a comparison of the 2D gels, and protein spots showing differential abundance are excised from the gel and are identified by mass spectrometry. So these 2D based approaches facilitated the identification of hundreds of plant proteins in a go and also helped in the identification of stress specific proteins. So no doubt that this 2D approach helped in the development of hundreds of proteome maps, yet this technique suffered from a number of drawbacks, of which the lack of reproducibility was a major concern. So in order to increase the reproducibility between the control and stress or treated sample, a 2D DIGE approach was introduced by Unlu and co-workers in 1997. So DIGE basically refers to the difference in gel electrophoresis and involves separation of control and treated proteins within the same gel and thereby reduce the gel to gel variations. Dyes technique involves multiplexing of two samples by labeling them with different fluorescent dyes, which are known as Psi dyes, and includes three variants including Psi2, Psi3, and Psi5. Of these, Psi3 is generally used to label the control proteins, Psi5 to label the proteins from the treated samples, and Psi2 is usually used as an internal standard that can be used for the calibration and normalization of spot intensities across control and treated samples. So the proteins labeled from these Psi dyes are pooled together and these are run on the same gel. So the difference in the spot intensities or protein abundance is done by examining the gels on fluorescent scanners such as Typhoon, and the differential spots are visualized as different colors as shown here. So this picture is actual image of 2D dyes gel of Helicobacter pylori showing these yellow spots that are specific to control condition and these red spots that are specific to the treatment conditions, and these blue spots are present in both the conditions. So these spots, the blue one are kind of uh, housekeeping proteins, and those are not altered in response to a treatment. Okay. So although this technique increased the reproducibility, it suffered from a number of drawbacks, such as the use of high cost instruments like this Typhoon variable mode scanner. Actually, its price was approximately 45 lakh Indian rupees in 2009, and use of sophisticated software such as Decider. So this Decider software, which is required for the identification of differential spots, this is also quite expensive, almost 7 lakh Indian rupees. So this approach although was very good, it could not make a huge impact in the field of proteomics because this was kind of very expensive and all the laboratories across the globe could not afford it. And in addition, we need some highly skilled manpower to examine these gels. So it was kind of a good advancement, but it was not able to make a huge impact in the field of proteomics. Anyway, so because of the development of these gel-based proteomic approaches, these majorly 2D were extensively utilized to investigate the plant proteomes. However, later and subsequently, and kind of slowly, we realized that these gel-based approaches majorly leads to the repeated identification of only high abundant proteins such as Rubisco and superoxide dismutase, or the proteins that are present in very high copy numbers. So high abundant proteins are present in much higher copy numbers, sometimes up to 1 million copies per cell, and can occupy up to 80% of the total proteome. And because of the presence of these proteins, the identification of low abundant proteins, which generally perform the regulatory functions and are crucial components of cell signaling, is quite difficult. So, in case you are interested in uh, identifying the biological response of some stress condition. So you are basically interested in identifying the signaling and regulatory related proteins. But those proteins are difficult to identify because of the presence of these high abundant proteins. These basically mask the presence of low abundant proteins. So this 2D gel is the gel of soybean seed proteins, and you can clearly see that the major fraction of this 2D gel is dominated by uh, different isoforms of seed storage proteins, including beta congolysin and glycinin. So because of the presence of these high abundant proteins, the number of identified and quantified proteins in a biological sample are way less than the actual number, thus highlighting the need to remove these proteins for increasing the proteome coverage of the plant samples. So Rubisco in green leaves and storage proteins in the seeds and tubers and roots are some of the common examples of high abundant proteins that hinders the identification of proteins which are present in the low copy number. So, therefore, a number of approaches were developed to get rid of these high abundant proteins in order to enrich the proteome with the low abundant ones. So, majority of these proteins were based on the selective depletion or precipitation of high abundant proteins such as Rubisco or seed storage proteins. Our research group in Korea developed a protamine sulfate based precipitation method for the enrichment of low abundant proteins from a variety of samples, variety of plant tissues, including leaves, seeds, tubers, and roots. So this was kind of a universal method for plant samples, and it's applicable to almost all the tissues. Uh, Increased its efficacy actually. So in addition to these precipitation methods, other approaches such as affinity-based enrichment of low abundant proteins were also introduced. In 2006, combinatorial peptide ligand library, commonly known as CPLL technology, was introduced by a research group of PG Righetti in Italy, and is available with the brand name of Proteominer under Bioret. So briefly, CPLLs consist of several million hexapeptides, which are prepared using 16 different amino acids. So these hexapeptides can recognize complementary amino acid sequence in a bait protein, and it can harvest it from the sample matrix. So in simple words, when the protein extract is loaded on a CPLL column under large overloading conditions, beads having affinity with the high abundant proteins saturate first, and therefore the major fraction of high abundant proteins are washed out due to limited binding capacity of the beads. However, due to low concentration of low abundant proteins, these proteins keep on binding with their partner beads when additional protein extract is loaded to it, and thus gets enriched. So these are some of the gel pictures showing the efficacies of developed methods for the enrichment of low abundant proteins. So these 2D gels represents the detection of low abundant proteins from proteins by protamine sulfate precipitation method. And you can see that selective depletion of this storage protein glycine helps in visualizing or identification of low abundant proteins, which are kind of masked by the presence of these proteins previously. So in addition, and specifically if you look at this gel picture, you can see a huge difference in the 2D gel pattern of before and after the enrichment method application. So the gel picture on the right, this one shows the human plasma protein profile enriched with the CPLL, while the left one is untreated one, and you can see how many new protein spots are visible after the removal of these abundant proteins. So once we have protein extract ready and we have enriched our protein extract with the low abundant proteins, next step is to digest these proteins to generate peptides that can be identified by mass spectrometers. So protein digestion methods available now are majorly categorized into in-gel trypsin digestion method and in-solution trypsin digestion. So during in-gel digestion, protein spots or bands are excised from the gels, are destained, reduced, alkylated, and digested by trypsin enzyme to generate the peptides. While in case of in-solution trypsin digestion, isolated proteins in the protein isolation buffers are directly subjected to trypsin digestion. However, again we realized that both of these approaches, uh, in both of actually in both of these approaches, the presence of residual salts such as urea, thiourea, and SDS that we use for the protein extraction and polyacrylamide that we use for the gel preparation of the proteins hinders the efficient protein digestion. Actually, when these compounds are present in the trypsin digestion buffer system, these can hinder or affect the trypsin enzymatic activity. So, because of presence of these residual salts and polyacrylamide, the digestion efficiency of only 40 to 50% could be achieved. So, therefore, to overcome these bottlenecks, relatively recently, a filter added sample preparation method, commonly abbreviated as FASP, was introduced. So, this method involves digestion of proteins in a smaller filter units, which facilitates the removal of salts and other interfering compounds from the protein mixture during trypsin digestion. And with the help of this method, and digestion efficiency of up to 90% can be achieved as visible in this gel picture. So in this gel, the first lane is marker, the second lane is having proteins dissolved in SDS, and the third lane shows the SDS depleted fraction with the help of these filters, and no visible protein loss could be observed in between these two lanes. So the fourth lane contains tryptic digest, in which only trypsin and digested peptides can be visible, and in the last lane, you can only visualize the digested peptides, and this is a kind of final elevate. So in the final elevate, only digested peptides uh, uh, can be visualized, and you can see there are no other intact proteins or even trypsin. So this FASP method was actually a game-changing method, especially for the gel-free or shotgun proteome analysis, and it allowed the increase of proteome coverage of different plant samples. So basically, if you are working with the plant samples whose genome is not sequenced, or a plant sample or any medicinal plant that contain lots of secondary metabolites, so these secondary metabolites can hinder the trypsin digestion. So in that case, this FASP method can be efficiently used to increase the digestion efficiency and overall proteome coverage. So coming to the gel-free proteomics. So for the quantification of peptides and proteins in the case of gel-free proteomic approaches, two independent methods have been developed. The first approach is label-free approach, in which the quantification of proteins is carried out either based on the peak intensity of the peptides or by spectral counts. That is, how many spectra corresponding to a particular peptide are formed. While the second approach is based on the labeling of peptides with isobaric mass tags such as ITRAC, ICAT, TMT, and celiac. Since these tags are isotopes and carry same mass, these do not alter the mass to charge ratio of peptides. And when these labeled peptides are injected into the mass spectrometer, these isobaric mass tags are ionized to produce the reporter ions, and quantification of peptides is done by the quantifying the reporter ion intensity. So recently, these labeled-based approaches have been modified tremendously to increase their multiplexing capacity, and since these label-based approaches avoid errors that arises because of different mass spectrometrians, these approaches turned out to be more reproducible than the label-free approaches. So in case of TMT, 10 such isobaric mass tags are available that allows multiplexing and analysis of up to 10 samples in a go. So these tags are actually quite effective if you are interested in performing a time-dependent study. So in that case, you can start with a zero time point or maybe a control. And if you want to investigate the effect of, let's say, heavy metals on plants, so you can investigate, like you can harvest the sample after 1 day, 2 day, 2 day, 3 day, and up to 10 samples. So up to 10 days, sorry. So and the beauty of these methods, isobaric mass tags or labeled based method, is you can label these with different mass tags, these TMT reagents, and you can, after labeling, you can pool them in a single as a single sample, and then you can run simultaneously in a single mass spectrometry. And so actually, because of this multiplexing capacity, the reproducibility of these methods are quite improved. Okay, so recently, just in last year, TMT Pro reagents were introduced that allows multiplexing and identification and quantification of proteins from 16 samples in a go in a single mass spectrometry run. So the TMT reagents basically consist of MS reporter group, spacer arm, and an amine reactive group. So this amine reactive group covalently binds to the peptide N-terminus or to lysine residues. And after labeling, the peptides are introduced into the mass spectrometer, where each tagged fragments during MS2, I mean tandem MS, I mean, or you can say second round of MS, to produce a unique reporter ions. And peptide quantification, as I told you previously, is accomplished by comparing the intensities of the reporter ions. So the development of both label-free and these label-based approaches facilitated even a deeper analysis of plant proteomes and allowed identification of thousands of proteins in a go. However, there was always a debate on the superiority of these methods. Some research groups they claimed that label-free approaches are better than the label ones due to reduced peptide complexities, while other claims the superiority of label-based methods. So in 2019, a comparative analysis of TMT method with a label-free quantification method having LFQ algorithm was carried out to evaluate their efficiency in terms of three different level of fold changes, including three-fold change, two-fold change, and 1.5-fold change across an entire data set. So in most of the cases, both of the methods achieved comparably accurate estimates of all the fold changes. However, this TMT method outperformed the label-free methods in the detection of smaller fold change, and that was quite true for 1.5 fold change. Okay, because, so this Superiority of TMT method was because of its higher precision and fewer missing values, suggesting that this TMT method is superior to the label-free method, and it can be aptly utilized in the plant proteome research to increase their proteome coverage. So, while working on these TMT-based approaches or any other tag-based approaches, the complexity of peptides is a major issue since proteins from different samples are pooled together. Therefore, the efficacy of these tag-based approaches are largely dependent on the peptide fractionation methods. So, this peptide fractionation is usually done inside the mass spectrometry during LC run, but sometimes we can also do the peptide fractionation offline outside the LCMS. So in our case, to increase the proteome coverage during TMT or label-free proteome analysis, we developed the peptide fractionation protocols that involves the separation of peptides in an in-house developed reverse phase column into 12 fractions. So this is how you can elute and combine 12 fractions, and we realized that application of this peptide fractionation method with in combination with TMT and label free shows good results. So in case of TMT, we could identify 7,000 proteins in a single go, while label-free analysis showed only 5,000 proteins. So this analysis or this comparative account also suggested that TMT uh, analysis could be a better approach for analyzing the plant samples. And in addition, um, uh, the the reproducibility of TMT approach was also higher than that of label-free approach after normalization, which further suggested a better suitability of TMT approach for the plant samples. Okay, so coming to the targeted proteomics. So as I explained in my third slide, Uh, targeted proteomics deals with the identification and quantification of already known proteins, and their major purpose is to validate the results obtained from the discovery proteomics. So, for example, suppose we identify uh, um, enzymes, let's say a catalase from the comparative analysis, and you hypothesize that this catalase could be linked to a disease and you want to validate its expression, but you don't have any antibodies against it. So what you will do in that case, these targeted proteomics approaches, including this selective reaction monitoring or multiple reaction monitoring and parallel reaction monitoring, can be performed. The aim of both of these approaches are same. However, these can be performed using specific mass spectrometers. These SRM and MRM can be performed using the Q-trap based mass spectrometers, while PRM can only be performed based on the Orbitrap based mass spectrometers. So these targeted proteomics, in these targeted proteomics, actually we know that to which proteins we want to target. So that's why the term is targeted proteomics. So in this, and these approaches are basically used for the validation of particular samples. So coming to the success stories in the case of plants, we have already developed the draft proteomes of Medicago and Arabidopsis. And interestingly, the number of identified proteins in both of these organisms were found to be almost 5 times higher than the number of genes that they carry. So this increased complexity of proteomes as compared to the genome and transcriptome was actually proposed long way back, and the concept of one gene one enzyme is no longer applicable anymore, and this was further found to be true after the development of these two proteome maps in case of plants. So it is now actually evident that one gene can give rise to multiple proteins because of various post-transcriptional modifications such as alternative splicing, RNA splicing, and gene editing, as well as various post-translational modifications. So at present, 400 different types of post-translational modifications have been identified that modifies the structure, location, or functions of target proteins. So for example, just the cysteine thiols of the proteins are known to be modified by at least 8 different kind of post-translational modifications, of which sulfinylation, sulfhydration, glutathionylation, nitrosylation, cyanylation, and acylation are most frequent, giving rise to different identity to the modified proteins. And it is also possible that there could be other yet unknown modifications on this amino acid which are so unstable that those could not be identified by the experimental approaches that are available as of today. So it's possible that we in future we could identify some more post translation modification on this single amino acid. So you can just imagine the amount of complexity in the proteome that arises because of the presence of the single amino acid on the proteins. So in order to entitle these proteins that are product of the same genes, the consortium for the top-down proteomics introduced the term proteoforms. So as per this terminology, proteoforms are the product of same gene that are differently modified and perform specific function which is different than other forms. Okay, so let's understand the concept of proteoforms with the help of this donut example. So let's assume this donut as a protein, and like the same donut can be modified in different ways by different toppings, each representing a contrasting color and flavor. The same way, a single protein backbone can also be modified to give rise to a new proteoform that performs a different function. So further, while the development of proteome maps of Arabidopsis and Medicago, we realized that only 80 to 85% of the obtained protein spectra could be matched with the genome sequence, and rest 15 to 20% spectra remain unmatched, which suggested that presence of some yet unknown novel proteins. And those, why I'm saying these as a novel proteins, because their information is not available in the genome, or their information is not available what we think the genome is. So this basically indicated that these proteins are not the product of known regions of the genome, and these can be translated either from the intergenic segments of the genes or any other regions of the genome that we previously thought of being redundant and non-coding. So we all know that in the genome there is lots of DNA that we considered as non-coding. So, but the recent proteomic studies have suggested that those non-coding part of the DNA could also code for the proteins, and that's why this 15 to 20% spectra remain unmatched. So because the genome information or the genome databases that we know today contains the information only on the coding genes. So therefore, recently the concept of proteogenomics was introduced, in which the integration of available genomics, transcriptomics, and proteomics approaches are used to track the origin of proteoforms or splice variants of the same gene. So in a proteogenomic approach, the genomic and transcriptomic data are used to generate the customized protein sequence databases um, that helps to interpret the proteomics data. And in turn, the proteomic data provide protein level validation of the gene expression data and help in refining the gene models. So these enhanced gene models can help further in improving the protein sequences databases for traditional proteome analysis. So I think this is my last slide, and so with this last slide, I would like to sum up my presentation. But before that, I would like to acknowledge the funding and support from National Research Foundation of Korea and Korea Research Fellowship, Kookmin University, my postdoc professor and now colleague Professor Sundar Kim, and my old lab mates. Thank you, and thank you for your patience.
Thank you, Dr. Ravi. Dr. Ravi, please try if you can switch on your camera.
Okay, just a moment.
You can stop sharing and then you can Thank you, Dr. Ravi. Now we can see you.
Okay.
All right. I thank you, Dr. Ravi Gupta, for your illustrious talk. And again, you started with the very basics of proteomics, and then you moved to advanced proteomics, that how proteomics has evolved over the three decades. And you have made us aware of several protocols and techniques that are available. And also, I love the explanation of proteoforms that you have explained with the help of donuts. Really, it's lunchtime in India, so
Yeah, so everyone
you
must
can
be
understand.
hungry.
So, Dr. Ravi, I means somebody has raised his hand. There are a few questions as well. So let me first allow who has raised his hand or his or her hand to interact with you. Just a moment, please.
Okay.
Yes. It's Dr. Abhishek Roy. Dr. Abhishek?
Yes, thank you, ma'am. Thank you, ma'am. Thank you, Dr. Ravi, sir, for a brilliant presentation. Just one small question, sir. Like you had explained SRM and PRM. So PRM was, I guess, for a technique in which we use all the ions or selected ions. I'm a bit confused on that, actually.
Yeah, so actually we during MS2 analysis we select the precursor ions that we are interested in analyzing in tandem MS in the second MS analysis. So that's why it's known as targeted proteomics. So in both PRM and SRM and MRM, we are basically looking for the change in expression of selected proteins, not all the proteins.
Oh, in both the cases, only selected proteins will be targeted.
Yeah.
Thank you.
Yeah.
All right. Dr. Abhishek, I think you have
Yes.
got your answer right. So,
Yeah, thank
Dr. Ravi, because as I mentioned earlier also, we have students also in our workshop. So one of the students, you have already answered this question, but somebody has asked, so it's my duty to ask you, which of the peptide quantification methods are better, gel-based or labeling approaches?
So, in my opinion, labeling approaches are better because we can actually, using labeling approaches, we can perform both absolute quantification as well as relative quantification. So, in case you are really interested in quantifying the presence of a particular protein that in terms of its weight or intensity, you can go for absolute quantification, and that can be easily done by the label based approaches. So label free approaches are basically for the relative quantification. So in case you are interested in analyzing the differential proteome, like how the expression or abundance of a particular protein is changing in response to a treatment, then it's better to go for label free approaches.
All right. Thank you, Dr. Ravi. Now, one more question is there. What are TMT Pro regions?
So TMT Pro reagents are basically extension of TMT, So previously, as I told you, that using TMT, we can only perform multiplexing of 10 samples in a go. So, but the TMT Pro reagents are similar to the TMT reagents. They are having the same mass, so that's why they are isobaric, and they provide additional 6 reagents. So you can perform analysis of 16 samples in a go. So because those are extension of TMT, so that's why they termed it as a TMT Pro.
All right. Thank you, Dr. Ravi. This was Dr. Ravi Gupta's session for today. And as ma'am has rightly mentioned, today was our Proteomics Day. So it is my duty now to thank Dr. Renu Deswal, Dr. Ravi Gupta, who gave us lectures today on proteomics. And also for hands-on session, I would like to thank Ms. Pratima and Ms. Shubhangi Kala. And with this, today's session is over and we will be meeting you tomorrow. I'm sorry, 9 o'clock for the session on drug discovery. Thank you very much. All of you can leave now and we'll meet again tomorrow, 9 a.m. Thank you.